Okay, here we are. It's the last day for lectures from the Computational Neuroscience group, and I'm really pleased to introduce Mike Stryker, who started it out with a PhD from MIT, psychology.
It's called Psychology and Brain Science.
Okay, so, but it was, I think it was one of the first neuroscience departments.
Yeah.
Even though it was called psychology. But then, and now it's, brain science, but he went to Harvard Neurobiology, worked with David Hubel, and a postdoc, and then directly, I think, to UCSF, and where he's been ever since. You'll see the work that he's done, classic work on binocular integration and the cortex. I learned just recently that he was actually responsible for a very important event in the history of computational neuroscience. Okay, so if you go back to the 80s, early days, basically you could go to a neuroscience meeting, see the posters, right? Literally tens of thousands of posters, and you might find one or two computational posters. It's basically, it wasn't part of neuroscience. Now it is. The important part of neuroscience. But there was a moment when the Sloan Foundation started a series of Sloan Centers for computational neuroscience. It was a time when there was a bunch of physicists who had lost their jobs, particle physicists, because the large collider that was being built in Texas actually was shut down. And so these very talented postdocs were available, and so the centers were set up, and we brought them in, and it was great because they had a chance to contribute. Turns out Michael's responsible for this. That's to say, it was he that got together with the program managers at Sloan and said that this would be really important for the field. And so thank you, Michael.
Chuck Stevens.
And Chuck Stevens, okay. Yeah. So Chuck Stevens, who's a colleague of mine for many years, was also involved. Yeah. Okay, well, Michael.
It worked. So it became a real deal. So this meeting is being recorded. So I'm going to talk about one form of adult plasticity, but I'll start just by reviewing some of the really old work we did, because
why is it stationary and running here?
I am displaying not the desktop, but my controls don't work. So let me stop sharing this and start sharing the screen. That's the desktop. And maybe this will make my controls work. There, it works. So a lot of the early work on the only behaviorally and neurophysiologically and neuroanatomically relevant form of activity-dependent plasticity was the work that David Hubel and Thorsten Wiesel did on the development of structures in the monkey, and initially they did it in the cat brain, that correspond in the visual cortex to input and concentrated responses to one eye versus the other eye. And this was a clinically very important problem, because if a human child has the vision of one eye occluded or blurred for the first 6 weeks of life, then vision in that eye is permanently compromised. And part of the explanation for that is that an organization where inputs from the two eyes go to alternating patches of about equal size in the input layer of the visual cortex is modified. And what happens is the inputs from the eye that was occluded become very contracted, and very few cells in the visual cortex outside of layer 4 respond to input from the deprived eye. And all of this was done with sharp little wires, microelectrodes, that could record one cell at a time. And so to record 30 cells took all day sometimes, because you have to isolate the action potentials from a single cell and then study it and see what made it fire. And this rearrangement we showed corresponded to, in about a week, axons from the lateral geniculate nucleus that convey the driving visual input to layer 4 of the visual cortex. Boy, very sensitive touchpad. That convey the driving input change from looking like this to looking like this. So the deprived eye axons lose their branches. And it was competitive. So if you blocked activity in the cortex with tetrodotoxin or just closed both eyes, you didn't see this rapid change. And this rapid change was associated with a change in which eye drove cortical cells. So, and this is all work that was done in cats and later repeated in monkeys. If you give a histogram of the balance of input between the two eyes, with the middle category being cells that were driven equally by the two eyes, and the extreme categories, cells driven only by one eye or driven only by the other eye. In 1 to 2 days, I got to get a mouse. In 1 to 2 days, this distribution of responsive shifts, so that hardly any cells are driven by the eye that had been closed for the last 2 days. And almost all the cells are driven by the eye that remained open. So this is a really dramatic form of activity-dependent plasticity. And you could do it with goggles, you could do it with a blurring lens. There was no invasive pathology. And when we looked at connections, and I'm not going to explain this experiment in great detail, but when we looked at connections within the cortex, between the two eyes, what we saw in the same period of time, the connections within the upper layers anatomically reorganized on a 0.5 millimeter scale within 2 days. So this is a very dramatic form of plasticity. And we worked at it, trying to figure out what the hell was going on, what were the mechanisms of this, what could we do to stop it, and so on. And it pretty much, I'd pretty much given up. I started writing a bunch of news and views in Nature on higher visual cortex in monkeys. And I thought there was no way we were going to make progress on this.
Progress in what sense?
Progress in the sense of understanding the mechanisms that were responsible for this form of activity-dependent plasticity, which we also hope would lead to a therapy for children, because amblyopia is still a real problem. Children who lose vision in one eye.
Michael, but that didn't stop you from modeling this.
No,
Having plasticity, right?
No, and in fact, Ken Miller, who's at the Theory Center at Columbia, was my PhD student, where we really made the first analytically detectable model of the development of ocular dominance columns and their plasticity, where the elements of the model were all corresponded to real things in the brain. And the parameters of the model could, at least in principle, be measured experimentally. So that was the start of Ken's career in modeling. And the modeling was great, but it was, for somebody like me, it was really frustrating that we couldn't go from models to molecules. And, I was ready to give up. And then mice came along. Yes.
So you show the synaptic radicalization is kind of what changes. Do you also lose neurons? Do you also lose neurons?
Neurons, the neurons don't die.
Don't die. Okay.
Yeah, we measured that. There's basically absolutely no cell death taking place. That's it. So what happened was it became possible to manipulate mice genetically. And so it seemed like with these genetic tools, it might be possible to dissect what was going on. And this is not the main subject of this talk, but it's saying, you know, even in the old days when we had single microelectrodes and it took forever to do experiments and we couldn't do very many by comparison with what we do now, we could figure some things out. So this data shows from Josh Gordon's PhD thesis in the lab, shows that in mice, as we knew in cats and monkeys and later ferrets, there is a well-defined critical period where depriving a mouse of vision in one eye for a brief period of time changes the distribution of responses to the two eyes. The reason this happens became clear when my former postdoc, John Kang, boy, I wish we'd done this before he left the lab, but he really revealed what's the reason for the critical period. I mean, why do we have this period of very heightened sensitivity? And the reason is that at the beginning, when the cortical cells are driven from the left eye and the right eye, they don't know about each other. So you will have a cortical cell which is selected for this orientation in one eye, and in the other, when you drive it through the other eye, it's selected for this orientation. After the critical period, after this 1 week period from about day 28, day 28 to day 32 or so, then when you record all of the cells, if they like this orientation through one eye, they like the same orientation through the other eye. And that depends on having visual experience during the critical period. So the rationale for the activity-dependent plasticity became clear. It's needed to match up the preferred orientations in the two eyes. Now, how this occurs is a huge shock and surprise. And it's another story that Josh Trachtenberg can tell. It's work that he did at UCLA, which I think is fantastic. But the other things that became possible were we could actually look at the synapses. So we could label the postsynaptic densities in spines, and we could follow them over time and see which ones were lost and which ones were gained. And that told us that the original effect of monocular deprivation is actually corresponds to a loss of input from the deprived eye. And we could dissect the various phases of monocular deprivation. So first you lose response from the eye that you close. Then the other eye, the responses get better, and surprisingly, the responses get slightly better to the deprived eye. And then if during the critical period, after a few days of deprivation, you open the deprived eye again, things go back to their original state of balance between the two eyes. And it turns out that we know the mechanisms, the mechanism of restoring the responses to the deprived eye is a neurotrophin-dependent growth of new synaptic connections, new spines and so on, from the pathways that serve the deprived eye. And if we block that using a chemogenetic genetic manipulation, we can completely prevent the recovery. It never happens if we block it, and so on. And this middle phase where the open eye responses get better actually corresponds not to a growth of new connections in the open eye, but to a form of homeostatic synaptic scaling, which we can block by tumor necrosis factor alpha.
Could you explain what that means, homeostatic?
So what happens is you take a bunch of neurons in a dish that connect with each other and make each other fire. They'll have a certain level of activity. If you measure the size of the individual quantum events, they have a certain size. You block synaptic connections, you block activity in this dish for 2 days, and you then wash off the tetrodotoxin that you block it with, or you can do it various other ways. What you see is that the individual synaptic events are twice as big. So this is a discovery from Rob Malenka's lab with David Stellwagen, who's at McGill now. But I thought it was silly. I thought it was a cultural artifact. So, but we did this in real mice, and the same thing happens in the visual cortex in vivo. And we can completely block this increase in the response to the open eye by blocking tumor necrosis factor alpha signaling pathways with a TNF alpha knockout or with soluble receptors that sell up the. But that's this is not the story I wanted to tell. This is just saying you can get a long way with a single microelectrode recording the activity of cells, but it's really painful and slow.
Yes, so mice are great because mice are great. You can do all these genetic manipulations, but mice, the mouse cortex is different from that of other mammals.
Yeah, it is. There are differences, but everything we know about the mechanisms of plasticity, when we, in a fragmentary way, test the same thing in ferrets, mostly not cats, because ferrets are like, a cat that's been through an extrusion mold, that they're the same. So mice are not men. And, I started talking about doing visual experiments in mice, and people would sing me the little song about 3 blind mice. But in fact, mice are very good predators. So Chris Neal's lab in Oregon has shown that if you put a little cricket in a cage with a mouse, actually you have to feed them crickets the night before. Once they've had a cricket, they'll never go back. And you put a little cricket in the cage, and the mice are very efficient predators at visually guided predation.
Yes, so you were showing this ocular dominance columns and
They don't have columns.
Yes.
They don't have columns for preferred orientation. They don't have columns for ocular dominance. And, I think it's an open question how much of what we learned in mice can be extrapolated to larger creatures. But at least at a mechanistic level, it all seems to be true. So we also...
Andres, they have antenna. So do they have columns or dominance columns in their antenna? No.
They have no dominance columns, no orientation columns. However, we believe that it's the same wiring, it's just not clustered the way it is. And we have actually quite a bit of evidence for that. But and in those days, we're always using anesthetized animals. And we did the standard sort of visual neurophysiology things. Chris Neal and I recorded the orientation tuning curves for excitatory cells and their spatial frequency tuning curves. So we found out with gratings and bars, what were the preferred orientations like? What was the spatial frequencies like? And we saw more or less as expected, the inhibitory cells had much poorer orientation tuning and much broader, but it didn't go to very high spatial frequencies. So it was as if the mice were seeing a very, very blurry version of the world. Maybe they had to be 50 times as close to something to see it as a cat would. So this is a summary of the data from our experiments in anesthetized animals. And life really changed when we could, for the first time, record from alert animals. Alert animals running on, standing or walking or running on a styrofoam ball that's floating in air, which is a rig that David Tank's lab used first for mice. I recall a discussion of Woods Hole with David when we were wondering how could we do alert mice, because he wanted to do them for his projects and I wanted to do it for mine. And he was very enthusiastic about copying Werner Reichert's fly on a ping pong ball device for mice. And they did it first, and then we did it to look at the visual system. And we were really concerned is all this stuff we see in the brain due to anesthesia? Will it really be the same in alert animals? And this was actually Bob Wurtz's first project after he had worked with Jim Olds. His first independent project was to see are the properties of neurons in the visual cortex that Hubel and Wiesel had found in anesthetized monkeys, are they also there in alert monkeys? And work showed that they were. And indeed, Chris and I, looking at alert mice, found the same thing, that there's orientation tuning, spatial frequency, and so on. However, the responses were really variable. And at first it was a mystery. And then we realized that if we separately considered the responses when the animals were standing there on the ball, fully alert, ready to eat a little piece of angel hair spaghetti, or when they were walking or running, the responses were qualitatively similar, but quantitatively really different. So the responses when animals were walking or running were much larger, but the orientation selectivity, the width of tuning and stuff were unaffected. So we referred to this state that's induced when mice start to walk or run on the ball as the high gain state of visual cortex, because it looks like just you're turning up the gain. Whereas when we looked in the lateral geniculate nucleus, which is the thing that provides the visual input, there was no such change with walking or running. So this was a regulation of the gain of cortical responses that did not affect their selectivity. So it's, I think there's a principle here for neuromorphs, which is that if you can separately control the gain and the selectivity or the specificity of the circuit, it's a real advantage. And so the question was, how does the brain do this?
Question.
Yeah.
This kind of thing has been observed also in insects.
Yeah.
Yeah. But when the mouse starts walking, it has to process faster. Not necessarily more gain, but it has to process more quickly. It has to decrease the time constant. That's different than the gain.
Yeah, that's an interesting.
It has to speed itself up, right?
Yeah, but it can work. I have something that'll address that in a little while. Yeah.
Are those responses from B1 independent from the environment? Meaning that if the mouse is moving but it's stationary, so there is no much more visual information that is going to his.
This is a mouse whose head is fixed.
Okay.
There's the visual environment's just stationary.
Okay, so there is no more visual information.
Well, the thing we obviously worried about is, well, maybe the eyes are moving, maybe there's something else happening. And that's why the recordings from the lateral geniculate nucleus were so important. Because if this was coming from the eyes, then you would have seen it in a pathway that provides the digital input to the cortex. So one of the ways, so there were two ways to approach this, either from the top down, from the cortex down, or from the legs up. And so in the legs up strategy, we found the midbrain locomotor center of the mouse, these had been identified around the end of World War II, actually, by some Swedes and Russians. That there's an area in the midbrain, if you stimulate it, it causes locomotion. And there are 3 kinds of cells in this area, and we transfected each of the 3 kinds of cells in separate experiments with an excitatory opsin, so that when you stimulate it with light, it makes the cells fire. And then we put a fiber down near them and turn on the light and look at the blue light above this mouse's head. As soon as the blue light comes on, the mouse starts to run. As soon as it goes off, he stops. Comes on again, he starts to run, and so on. Yes, this was indeed the midbrain locomotor center.
So do you think the mouse is aware that it's running because you're stimulating it, not because it wants?
No, we weren't able to read his diaries that he wrote that. So the question, and of course, when the mouse started to run, he went into the high gain state in the cortex. I'm not showing that. But that was completely uninformative, because even if we hadn't stimulated the midbrain locomotor center, when it started to run, it would go into the high gain state. What was informative, though, was we could turn down the intensity or the frequency of this stimulation to a level that the mouse didn't walk or run anymore. So the mouse just was stationary. But when we stimulated like this, the mouse still went into the high gain state. So we speculated that it was not the descending pathways that make the legs move, but the ascending pathways from the midbrain locomotor center that was responsible for going forward into structures, closer to the cortex that put the cortex into the high gain state.
So
Going the other way, what?
is that a switch, or is this
It
have a
seems
dose response?
I mean, is it binary, the two
Yeah.
states? At least with walking or running it is, but I don't think I think it seems to be, in all the circumstances we've investigated, I don't think it would have to be. And it's, anyway, so to go in the other direction from the top down, we looked at what are the different classes of cells in the mouse cortex to which we have genetic access. So we could do essentially the same experiment we did with the midbrain locomotor center, but with one cell type in the cortex at a time. And surprisingly, there is a minor population of interneurons, of inhibitory neurons in the cortex that express vasoactive intestinal peptide, which, fortunately for us, they're known as VIP interneurons. And when we turned on the VIP interneurons, we saw two things. First, when a mouse is normally alternating between standing there and walking or running, this is a plot of velocity of the mouse on the little ball versus time. If we looked at the amplitude of a calcium signal from these VIP interneurons, where we had put a fluorescent calcium indicator in them, what we saw is that it paralleled the mouse's locomotion. So whenever the mouse started to walk or run, the VIP cells were active. Whereas when we looked at most neurons, all the other kinds of neurons, there was no parallel to the walking or running. So it was sort of a mystery, because the cortex goes into a high gain state, but the neuron that's responsible for it is an inhibitory neuron. So how could this work? And fortunately, our colleague Massimo Scanziani and Rafa Justi had essentially the same story. The output of the VIP cells goes to another class of inhibitory neurons called somatostatin cells. And somatostatin cells provide a very powerful input to the dendritic tree of the excitatory cells in the cortex. So when the VIP cells are active, they turn off the somatostatin cells. The somatostatin cells no longer inhibit the normal excitatory cells, and the excitatory cells fire more. And somatostatin cells are not a single class, but we could find many somatostatin cells whose activity was the opposite of that of VIP cells, consistent with this when we recorded their activity in relation to locomotion. So this made real sense. And the VIP cells are really interesting because they're near the top of the cortex. They get lots of input from higher cortical areas, and they project straight down in a little column onto the excitatory cells under them. Yes?
I'm not sure if I remember correctly, but I remember that somatostatin cells can also do lateral division.
Yeah, somatostatin cells do lateral. VIP cells do not.
Would you expect then the receptivity to become less specific?
No, because the driving input, the excitatory input, which is really responsible for most of the specificity that these cells have, is unaffected. So it's this separation of the circuit that this one circuit is responsible for the gain by having various amounts of inhibition. And the excitatory circuits that are responsible for the selectivity are unaffected by this. So, that's how I think it is. So, if this were really true, we ought to be able to excite the VIP cells in a mouse that was just standing there and get the same effect we got with running. And indeed, that was true. If you transfect the VIP cells with an excitatory opsin and drive them, then you get an increase in gain.
So Mike, how does this high gain affect what the mouse sees? Does it make them more visually sensitive?
That's the part of the talk that's not the introduction.
Okay. I'll wait. I'll come to that.
Yeah, so it's very hard to prevent cells from firing with most of these tools. So after trying 5 different ways of suppressing the activity of the VIP cells, we just blew them away with, focused a laser on them one by one and destroyed them. And, in these experiments, before we destroyed them, we saw this big increase in gain when the animals started to walk or run. After we destroyed the VIP cells, it didn't change the specificity of the responses at all, but we got no increase in gain when the animals started to walk or run. So this, I think we had, loss of function and gain of function experiments showing the VIP cells. So what was really interesting to us, does the cortex have special plasticity in this high gain state? And the first experiment we did about the plasticity and the VIP cells had to do with the recovery from amblyopia. So if we do this model like a human child who's had his eye occluded for 8 weeks, and we can ask, how do the responses, do the responses to that eye ever get better when the optical problem that occludes that eye is removed? And the answer is yes, they get better. But in mouse and in human kids, they only get about halfway to normal. But what we did was to take this model in mice and instead show the mice a particular visual stimulus during actually for an hour a day, while the mice were recovering after opening the deprived eye and after the end of the critical period. And, if the animals were just allowed to run, nothing happened. If they were in their home cage, that was the control. But if we gave them a particular visual stimulus during locomotion, then what we saw, we could recover responses that were within the normal range. So this suggested that the high gain state can really influence plasticity. So we had, several stories that running enhances responses, that the VIP somatostatin circuit is activated by running to enhance responses, and that running enhances plasticity. But of course, that doesn't say the plasticity is coming from this circuit. Maybe it could enhance plasticity directly without running, or maybe running is aerobic and just, like Shihab, it keeps you looking like a god for the rest of your life. and makes you really smart, has plasticity. So I thought of
You
Shihab.
probably know that in audit, but not probably, I'm sure you know that the auditory does the exact opposite.
Yeah.
So everything you described here is so basic to the circuits in the cortex. What is different about the auditory?
I don't know. That's a long discussion we could have later. I have a speculation, but it's true. In the auditory cortex, responses are not enhanced by activating the VIP cells. Yeah.
So I don't understand why you need to add visual stimuli plus running, because when the animal's running, doesn't it see stuff normally?
Well, the animal's running in the lab, it doesn't see anything because it's just the stationary lab out there.
Okay, so
it's
did the head fix.
It's a running head fix. Okay. So basically, to address this question, whether it's really the aerobic activity for running that's good for plasticity or something about this circuit, we used a tetanus toxin light chain transfection of the VIP neurons. And so this prevents them from releasing any neurotransmitter, either VIP peptide or GABA, both of which they release. They can't release this anymore when they've been transfected. The orientation tuning of the cells was unaffected, but it completely blocked the plasticity that was gated by locomotion. I won't go into all the details of this, but if the VIP cells couldn't release anything, then it didn't matter. It didn't help the plasticity. Whereas if we optogenetically activated the VIP cells, we could get plasticity just as if the animals were running. Yes.
Is there any other more activity in the neurons that can induce the kind of thing?
So I think, let me defer for a little bit.
Okay.
Okay, because that is really the question. What else drives these VIP cells? So fortunately, when we're doing this experiment, in some experiments we used contrast modulated noise like this. And in other experiments, we used gratings as the stimulus that the mouse watched while it was running to induce plasticity. And this turned out to be a really fortunate accident, because when we compared the effect on visual responses of showing animals a visual stimulus for an hour a day while they were running, it turned out that the enhancement of the response was entirely stimulus specific. So if the animals saw the contrast modulated noise when they were running, and we tested them with contrast modulated noise, then that response got much bigger. But the response to bars or other stimuli didn't change at all. And that was true no matter which stimulus we used. So this was a stimulus selective enhancement of response. And it lasted for a very long time. It lasted for at least 3 weeks. And so this is just more details of the same thing, using different stimuli to be shown while the animal was running for an hour a day. And you produce this enhanced response that was specific for that particular stimulus. And it turns out 1 hour a day was saturating. If you gave them 2 hours or 4 hours a day of experience, it didn't change. And locomotion was crucial, because in the same rig with everything the same, except turning down the air so that the styrofoam ball made it too hard for them to walk or run, the animals there was no enhancement of response, whereas, in the previous experiments, there were big enhancements. So locomotion was essential. And when we looked at the properties of individual neurons using 2-photon calcium imaging, what we found is, if you look at this row D at the bottom, the original tuning of this neuron to the different stimuli indicated above was like this. So there were one orientation in either this direction or that direction produced a pretty big response. But after we had shown the animal this 45-degree bars moving back and forth, that response got very much bigger, and the other responses were unaffected. Whereas if we, in the control case, if we didn't show them anything, or if we picked a neuron whose responses that didn't respond to the stimulus that we were showing during locomotion, its responses were also unaffected. And so, in some experiments, we did 45-degree lines or 135-degree lines. And, the appropriate one was hugely increased and the others were not affected. So I'll go through. So what's special about the high gain state? It's special that it can induce this stimulus-specific plasticity that lasts for at least weeks. It's not clear why this is useful, but one question is there more information in the response of the visual cortex when the animal's in the high gain state? And Maria Dotterlot, who's at Purdue now, did this experiment to see it, where she measured the modulation of responses of a large number, usually about 60 to 100 neurons recorded simultaneously, because our technology had advanced, you record from lots of neurons at once, and looked at the changes in their responses. And if we looked at the stimulus-specific information produced by locomotion, indeed, there was a big increase, particularly in the upper layers. And it really did not depend, it wasn't just the fact that there were more spikes, but there was more information per spike about the direction of the stimuli that the mouse was looking at and about the orientation. So the high gain state had more information. And in some ways, this is partial answer to your question, Shihab. If we looked at how much information measured by the classification error with a linear discriminant analysis between the spikes during locomotion and not during locomotion in individual experiments, if the mouse gets, if the error fraction is like this at, for the first 100 milliseconds of response, and of course, if you have a longer period of gathering spikes, the error fraction gets smaller and smaller. But say the 100 milliseconds gives you this much information about the orientation of the stimulus. If the animal is stationary, you need 300 milliseconds. So the increase in information per spike plus the increase in the number of spikes makes a huge difference in how fast, at least using this linear discriminant analysis measure of the information that we can decode from the responses in the visual cortex.
So
it seemed to be really useful, and that makes sense, because if you're going to have heightened plasticity, you want to have heightened plasticity in a circumstance in which it's plasticity that's encoding something real about the world. Yeah.
So LDA in time, you're running linear discriminant analysis in time.
Yeah.
So you're presuming some window that you're doing the analysis.
Yeah. The window is either 100 millisecond window or 300 millisecond window. So the longer you accumulate spikes, the more information there is.
Yeah.
But we're saying if you just look for the first 100 milliseconds of response, how long does it take to get that same amount of information if the animal is stationary? And it turns out it's 300 milliseconds. So this made sense then. Well, if there's more information, it makes sense you want more plasticity. Then we asked which of these two things that VIP cells release is responsible for the plasticity. Because people had long thought that these peptides, which had long-lasting actions, would really be important signals for plasticity. So we separately knocked out the release of GABA, the inhibitory neurotransmitter, from the VIP cells, or knocked out peptide, which kept it from releasing peptide, but it released GABA normally. And what we found in one of these stimulus-selective enhancement experiments is if you knock out VIP, the peptide, it looks pretty normal. Whereas if you knock out the GABA, the cells are perfectly happy. They look completely normal histologically, but they fire normally, but they just don't have any effect on plasticity. So it seemed like it was the activity of those cells. And, this is something interested me. Is this stimulus-specific enhancement still present in old mice? And the answer is it's a lot slower in old mice, and it doesn't last as long, but you can still get it with twice as long an exposure. So I asked the question that really interests me. What is enhancement for? I mean, running and walking is very convenient for us because we don't have to ask whether the mouse is paying attention. It just turns on the circuit automatically. But presumably, the widespread input, top-down input that VIP cells get from other areas in the cortex, presumably when the mouse is paying attention to something, also might be the thing that normally gates their activity and gates plasticity. But of course, mice do have to pay attention when they're walking or running. So this struck me as something like perceptual learning. And is this circuit the circuit that is the one responsible for engaging perceptual learning?
So Mike,
And yeah,
I mean, so there's like a whole other story with some similarity relating to kind of pupil dilation and brain circuits that
we showed that the pupils dilate when the animals start running, which indicates there's norepinephrine.
Yeah. So how do you think those two circuits?
I don't know. We haven't had the tools to take that apart, but they may co-occur. Yeah, I just don't know. There are other neuromodulators, but merely causing the VIP cells to release GABA is all you need to do to get this.
Do you know if like pupil dilation without running? I mean, first of all, does that activate VIP? Does it cause eye gain?
Pupil dilation is AC coupled to running. So the VIP cells stay up and have high level of activity as long as the animal's running. The pupil dilates at the beginning of running and then it gets smaller. So when we look at the relationship of locomotion to VIP cell activity and to pupil dilation, it's much tighter correlation with the VIP cell activity. Yeah.
You get it further in you get a natural student effects.
I can't hear.
You get a further change if you actually, instead of that complex mode that you actually have if you get natural sleep.
I don't know. I don't know. But I'll show you the experiment we're trying to do. So, what is visual perceptual learning? It's like, for those of you who are not Germans, when nowadays we learn German, we learn it in normal Roman script, but of course it is also written in Fraktur in Gothic script, right? And I know when I was learning, it was really hard to read that. I'd have to do it letter by letter for quite a while. So, how many of you can read this? Pack up your robot in Gothic script. But after perceptual learning, you just read the bottom line and the line above equally easily. And that's what perceptual learning is. So we're interested is this VIP cell circuit involved in perceptual learning? So what we created with Nikhil Bhatla, who was a postdoc, is a head-fixed virtual reality environment for mice, where we could make recordings as well as have the mice do things. And this is an example of the video game the mice play. There will be two trials. The mice are trained to run toward a cylinder with horizontal stripes and distinguish it from a cylinder with stripes at a different orientation. And we take pictures of the mouse's eyes. And here's a picture of the VR setup. And this is the mouse that's not super well trained, and he runs toward the wrong one and gets a little timeout. And then in the next trial, he runs across the grass toward the right one, and he gets a drop of water. So, and actually now we give them sweet soy milk. So, it lets us study visual perceptual learning. So the experiment is to train the mouse on gratings that are rotated clockwise from horizontal until the mice can do a very difficult one at 15 degrees. And then we can test them with 45 degrees, which is easy for them, 15 degrees, which they, after a month of training, they can do, minus 45, rotated the other way, which is very easy. They can do it immediately. And then minus 15, and they can't do it. And so this shows the result using D prime as a measure of discriminability. And basically they're pretty good with the easy ones. They're pretty good with the trained ones, but they really can't do the untrained one at all. So then our strategy is to kill off the VIP cells in the visual cortex, which I'm sorry, I don't know if you can see, but in this area where we injected a caspase that selectively kills VIP cells, there's a dark black area in the middle of this picture, but it should be on a black background so you can see it. There are no, the VIP cells are gone, but if we use a stain that makes the VIP cells red, you don't see any red cells here. But the rest of the neurons are just fine. So we're very selectively killing the VIP cells. And then the experiment is to try to retrain them at the minus 15 degrees. And in a mouse where we haven't killed the VIP cells, they can be retrained, and it takes about the same amount of time to do minus 15. And we're in the middle of the experiments in the animals without VIP cells. So we think that, you know, the system is present in all mammals. It's widespread. There are cholinergic effects that have been widely reported. And the input to VIP cells, the driving input to them, is very strongly nicotinic acetylcholine. So we think this system may be really important for perceptual learning, but we haven't shown it yet. So yeah, I think that's all. This is the crowd that's done these experiments. And in particular, Chris Neal did the early recordings and the discovery of the effect of locomotion. Yu Fu is the one who identified the VIP cells. Moses Chow did the midbrain locomotor center, and Muzumi Kaneko did almost all the plasticity experiments, and Maria Dodrobot did the ones measuring information. Thank you. Okay, that's quite a tour. Questions? Yeah, heard.
So I learned that we want to stay agile, and we should all go for runs in Telluride with beautiful contrast.
I think so. And some of my Italian colleagues. So we were very interested to see whether we could detect this in the human cortex, detect the high gain state. And, you know, there is a high gamma that's 60 to 100, something, you know, above 60 hertz. And we were just unable to measure it above the noise when you put people on a treadmill. But some of my Italian colleagues put people on a stationary bicycle and measured changes in perceptual learning. And it was not, you wouldn't bet your life that this is true, but it was a cute experiment that it was consistent with what we found. But
so forget about the treadmill.
It's a nice story, but there is the mystery, which she pointed out, of why is it in auditory cortex that the responses don't get stronger during locomotion and when the VIP cells are activated. And the hand-waving explanation I can think of is, first, the eighth nerve systems have very high spontaneous activity. The average spontaneous activity in the upper layer neurons in the visual cortex is about 0.01 hertz. So they rarely fire unless there's just the right stimulus there. Auditory cortex is completely different. They're firing a lot. And it may well be that in terms of information, it's better to shut things down in the auditory system rather than to turn things up. But this is just bullshit, right? Yeah.
So the paradigm that you started, and people tried in the auditory, as you said, that produced suppression rather than testing, has become a really nice standard paradigm for studying predictive effects. And it's basically when you have sensory motor stuff. And so I wonder if you have looked at this angle in the visual.
So what will we do exactly?
Well, what they do is they, for example, have the mouse run, and then they look at the influence of the motor signals into the auditory cortex and how they produce suppression.
Yeah.
And you can make it more precise. For example, with the running, you can't really separate the movement of one limb from activity, but you could, for example, have well-defined movements, and then you can see the predicted responses and the other, and it's associated with auditory. So I wondered whether the visual must have.
It's an interesting, it'd be interesting to do. I encourage, but I mean, these experiments can be done now. Originally it was hard to get the right genetically altered mice and all this stuff, but all of the apparatus and stuff for this is published. So I encourage anyone who wants to go into neuroscience and do this. That's a great experiment.
Yeah, I'll just quickly mention, Georg Keller's done a whole series of experiments like this in visual cortex with kind of the influence. So the idea, as the animal runs, he has the virtual reality screen move proportional to the running.
Yes.
And then it sets up a kind of predictive coding situation. And he's got a whole bunch of papers about it, but he's never looked at how VIP models and how they're related to any of that.
But I don't ever remember mentioning the upstate, downstate.
He's in all of his data, like running is let's see. The simple thing to say is I don't think he's ever looked at VIP neurons one way or another.
So
Yes.
Yeah, go ahead.
Yeah, so the Stryker movement where VIP was inhibiting.
That's that.
It's also inhibiting this parvalbumin cell.
Yes,
what's
but-
called that?
So the parvalbumin cells are all over the map, how they relate to locomotion. So yes, it inhibits parvalbumin cells, but
The cells inside.
and the circuit is complicated, and I wish I known about Gerald's stuff, because we might actually have looked at the dynamics of that. But the problem is usually we can label one kind of cell with a calcium indicator in those days. So we know what that kind of cell is doing, and then we know all the other cells, and we don't know who they are. So it's harder. So we did the VIP cells instead of setting cells separately. Now there are a number of different colors of calcium indicators. So one could actually do this and try to understand the dynamics of the whole system, which we can only innovate about now. So, but I think that's great. We should really do that because we've been able to get the JY count, the yellow fluorescent thing to work recently. Yeah.
You promised to say something about any other both inputs to the VIP or other motor acts that create.
Yeah, so all we know is that the VIP cells are among the cells. They're not the only cells, but they're among the cells that receive long distance input from places like ALM and from mouse frontal cortex, and very widespread inputs come to the VIP cells. And but not only to the VIP cells, but they essentially come to layer one and the area between layer one and layer two, where the VIP cells arborize very densely. So, so what I think is that in normal life, when the mouse is paying attention to something, I mean, I've never done it with the crickets, but because virtual reality crickets don't work all that well. But I'll bet these VIP cells are really just turned on and the cortex is in its highest gain state. But so I think it's probably, it just turned out to be convenient for us that walking or running turns the system on. But I don't think that's the only thing that does it in natural life.
So one last question. Okay.
So is that the equivalent of the amygdala for the mice?
What?
Is that the equivalent of the amygdala for the mice?
I don't think it's still mice have an amygdala.
Okay, they do have.
Yeah. No, I think it's I think it's the system that turns on the cortex to its highest activity and metabolic state, and it's probably too metabolically costly to be in that state all the time, and so to be able to gate it makes a certain amount of sense. But again, that's hand waving.
So I just have one last comment, which is that there's a big literature on perceptual learning that has nothing to do with running.
Yeah.
Right. But there's a reward, obviously, because you do have to train and reward if you can see the difference. And it also is associated with changes in the tuning curves.
Yeah, there are a number of things that people have called perceptual learning that I don't think meet the strict criteria for perceptual learning, that the same amount of experience with and without, engaging perceptual learning will either have an effect or not have an effect. But yeah, there is. And, it may turn out that we're completely wrong about, we're in the middle of these experiments. So I don't know.
But my guess is that maybe reward learning puts the cortex in the high gain state. Reward-based learning, where you're trying to achieve a goal.
Maybe.
The dopamine, right?
Yeah.
Well, why doesn't it just perceive clearly and reward by itself? Why does it have to be rewarded by some just perceiving clearly and sensing clearly is rewarded by itself?
Yeah.
It doesn't have to be simulated by some task.
So this is describing the experiments that had been done, how they did them, right?
Okay. Yeah.
In other words, it could also be just
Intrinsic reward.
intrinsic. Yeah, that's right. Okay, let's thank Mike. What's yours? Steve and I actually initiated, this is an unbelievable number of years ago. You calculated it.
Do not calculate it.
40 years ago.
Do not calculate it.
But Steve has really an extraordinary career in areas of like computer vision, computation. When I first met him, he was doing relaxation computation, and that, of course, is similar in flavor to what I was working on, which is the Boltzmann machine. So, but he's, as you'll see, he's very geometrically, he has a great geometric intuition, and he's now using it, which I think is a very powerful way to analyze neural data. And it's also going to be tied in with some of the work that Mike Stryker has been doing. So
Thanks for that. So it's a delight to be back here in person after COVID. It feels really terrific. And I'm going to tell you a story that took about 5 years to unfold. So it's going to go mostly in terms of driving the intellectual thrust of the project. And as you'll see, it was not only our computational group, but Mike and several other people involved in collecting neuroscience data. And it leads to this huge issue of how to begin to think about processing data so we can make inferences about functional neural circuits from the current perspective. Now, there we go. So why should all of you neuromorphs pay attention to this? As Toby said yesterday, the people who developed deep networks did so primarily from a motivation that came out of very basic neuroscience. And this is, of course, the original Hubel-Wiesel model for lateral geniculate cells feeding into simple cells and simple cells feeding into complex cells. And this notion of these feed-forward multi-layer circuits, what were called Pascal yesterday, I believe, have been a huge motivation for not only the deep networks, but this is Fukushima's original model for doing this. And of course, the history here is remarkable, where Fukushima worked for the post office in Japan, and we all love MNIST. This, of course,
that was the Buffalo post office.
The Buffalo post office, yes. And so that would be Jan. And in addition to, as Toby put it yesterday, Jan not being so willing to admit the intellectual foundations of these networks in the current context, many others have begun a small Manhattan project where they claim that in the spirit of theories of the brain should be coded in terms of the latest technology, that deep networks are the right machinery for thinking about how the brain works. And Jim DiCarlo, Danny Amons, I mean, lots of people. This is a huge Manhattan project going on,
But not nearly as powerful as the Manhattan.
not nearly as powerful as the Manhattan Project. In the title to this slide, I put question marks. Are these deep networks really good models, appropriate models for visual systems, despite this ongoing Manhattan Project?
Well,
So
we know they're not. We know they're not, but.
please shut up. Thank you very much. Thank you very much. Okay. So with that said, how can we go about addressing this kind of question? How is it possible that we can begin to do things? So like Mike showed you, we have a stimulus on a screen. We place some electrodes into the cortex of this subject animal, and then we do some analysis of the data. And the question, there are many levels of question here. One is, should we use mice? And Mike addressed a lot of that. What stimulus should we use? How do we analyze the data? And so we're going to address all three of those in an increasing sequence of complexity. Okay. This is the picture, or this is a version of the picture that Mike showed from his classic paper with Chris Neal, where they first showed the orientation selected responses in these beautiful receptive fields in mouse visual cortex. And as you can see from this, that the peak of these cells that were found were tuned to extremely low spatial frequencies. And I believe Michael even is still using Gabor patches centered around.
So those who can't read that small font, it's 0.04 cycles per degree.
Yeah, that means receptive fields are 10, 20 degrees in visual diameter, right? So these are huge. And to give you a feeling for it, if you think about primate acuity, you would see something like this. If you were looking through filters of that spatial frequency range, you would see this big blurry mess. So a lot of the primate forward visual scientists like Tony Mopchin published statements that said, well, the mouse is just not an appropriate creature. Why study vision in, as Mike put it, 3 blind men? And the logic here is, of course, it's all about receptive fields, and these receptive fields look like filters. And if that's the case, then it's really these low spatial frequencies. It's all about filters. But there's all this other stuff that Mike just talked about. There's all these beautiful molecular and genetic tools. There's all this beautiful imaging that's available in the mouse and no other creature. And so what are the real capabilities of the mouse as a visual creature? If we look at just the structure of the neuropil and things, well, mice aren't all that different from primates. There are certainly some differences. If you read the connectomics literature, it's not actually as severe as one might seem. So maybe the problem with the classic approach is we should not be just using visual gratings as the stimuli. Maybe the problem is we've been fooling around with the wrong stimulus. And so what's the day of a mouse in the wild like? And this gets to your point about somewhat naturalistic stimuli. Well, we look at mice outside of New York City, and basically they love to run through fields. As the mice are running through a field, they see this kind of waterfall of shrubbery and grass and stuff flowing past.
They're only running when they're trying to escape from being caught, right? The rest of the time they're foraging.
Well, they're still moving, right?
Yeah.
So there's still this waterfall. It may not be perfectly consistent, but they're still moving through. So there's this kind of waterfall of structure available to them. And we, being mathematicians, we like to take this complex natural beauty of the field and abstract it down to what we call these flow patterns. And so we've been experimenting with these flow patterns with Mike, and I'll show you some of that stuff for a while now, and they're really, really quite remarkable. You can adjust the contrast, you can adjust whether or not there's orientation information in the flow patterns or not. And here are some examples of what these flows look like. So you can see they're jiggling a little. We did some sexy computer graphics on this stuff to make them not completely rigid, motion, etc. And for those of you in experimental labs that would like to use these things, they're available.
Wait, Steve, I'm getting a percept of shape. Because it looks like there's a low frequency bump.
A lot of that's the zoom artifact.
Yeah.
And also, how come you don't have a focus of expansion?
This is the beginning of the project.
Okay.
Yeah, this is the beginning of the project. So we think of these flows as being a kind of intermediate structure. They're more naturalistic than the grading patterns, which we saw don't work very well, but they're much more able to be analyzed in ways that you'll see in a few minutes than natural images. So we think they occupy a kind of sweet spot in between. And in order to test whether they're actually appropriate for mice, this is where the collaboration with Mike's group began. And you already saw his setup with the fixed head mouse, awake behaving, running on the David Tang trackball, etc. And uses these 128 channel probes, etc. And what kind of structure did Michael find in these recordings? He found that, well, most cells in V1 mouse prefer some mixture of all these different kinds of stimuli. And I'll show you the exact ensemble shortly. A small percentage like the flow patterns only, small percentage like the grading patterns only. Most of them were really quite mixed. So among these cells that are giving these interesting responses, here is a spike triggered average receptive field at about 0.04 cycles per degree. So this was that optimal amount from the Neil Stryker paper. Now, if you look at some of our flow patterns superimposed on receptive fields of this size, you see that there are zillions of elements inside each of the lobes of the receptive field. The filter model just doesn't make any sense because they would average away to zero. There's got to be something more going on here. So this takes our opening picture, makes it a little bit more complicated, right? Instead of probing with a stimulus, now we have an ensemble of stimuli, and we get over the spiking activity. How should we process that kind of data? What's the best way to do it? Do we build tuning curves, all the classic stuff that you all learn about. I lost my ability up. So, all right. I should not have told you to do this.
That's fine.
No, so all the slide build-ups now stopped working. Anyway, it doesn't matter. Instead of introducing my piece, I'll walk you through it, okay? So here's the way to think about it now. In the new setup, each neuron that is recorded from sees some number of trials, stimulus A, some number of trials, stimulus B, et cetera, et cetera. So for each neuron, there's a whole mess of stuff. And what people typically do is they say, well, this neuron here responded to this trial this way. So they make a plot where this is the response of neuron 1, this is the response of neuron 2, ... This is the response of neuron 100 to this grading pattern, to this flow pattern, etc. And so they express trials in terms of neural coordinates. Well, it's hard to see in 100-dimensional spaces, so you PCA this 100-dimensional space down to a few dimensions, like 3 here, and this is the classic way to approach this. There are now, what would you say, Terry, 30 papers, 40 papers a year? 300 papers a year.
PCAs.
Yeah. Everybody's
the standard.
Yeah, so Joe had these pictures in his talk. I'm sure other people had them in their talks here. But basically what you see is a representation of trials in neural coordinates.
And what are the colors?
The colors here actually are going to correspond to the different stimulus ensemble, but I'm going to build this up a little bit more slowly, okay? We call this the encoding approach because companies like Neuralink really want to use this to figure out what you're looking at. So they want to put an electrode in your brain or a multi-electrode in your brain, take the output of that, and from that infer what you were looking at. And that's what this thing is, right? Each dot here is a trial. So you can decode what the animal was looking at by sampling and running your classifier or whatever on this, in this space of neural responses, okay? This is a great thing to do, and as Terry indicates, 300-400 papers a year are doing this sort of thing now. However, from our perspective, what this solves is which trials are similar, which trials are close to each other in this manifold of trials embedded in neural coordinates. Yeah.
Kind of seems to be you're going the wrong way about it. I mean, to me, it seems like you should have presented random stimuli, and then do the PCA and then select from there the stimuli that actually.
Zillions of people do that sort of thing. You can't get enough random stimuli to capture the power of these flows. It's so that these systems are so nonlinear. And so in slower versions of the first 3 slides here, I would have talked about that.
I see. Okay.
Yeah. So we think the community is working with the inverse of what they should be working with. That is to say, instead of asking which trials are similar, we want to know which neurons are functioning the same way, which neurons are similar. So we want a manifold, not in which trials are embedded in neural coordinates, but we want a manifold in which neurons are embedded in coordinates that relate to both the stimulus and the response properties, okay? So this is, in some formal sense, like a pseudo-inverse of what we're looking at. We would like to know, once we have these manifolds, which neurons are similar. So we would like this manifold to be one where neurons that are responding similarly to our world are near each other on the manifold. Neurons that are doing something different with respect to the world are far away from each other.
How is that different again? I still don't quite get it. Why is that different?
Here, each point on the manifold is a trial embedded in neural response coordinates, okay? Here, each point on the manifold is a neuron embedded in stimulus coordinates.
Stimulus response.
Stimulus response coordinates. Okay, so each point here is a neuron, each point here is a trial. Okay. Okay. It's going. I'm going to do this twice, Toby, because it's
Where did the trials go? You've lost the trial.
You'll see. So they're going to be embedded in the ambient coordinates here.
Okay. So you still got them.
Sorry.
You still got them. You're not representing them.
That's correct. No, they're implicit in the ambient space. Yeah.
You can see that you can do this. You've got this parameterized to generate the stimuli. So
No. The algorithm will give you will work on anything, right? And our stimuli are actually an ensemble, so we don't have a parameter that runs over them, right? It could be a discrete ensemble. Yeah. And there is no single parameter that runs over them.
Interpolate between them somehow.
Yeah.
Can you just say a little bit more about like why you want to do this?
Yes, I want to know how neurons relate to each other, not how stimuli relate to each other. I'm going to do 3 examples.
Yeah.
So this.
So I mean, like the brain doesn't obviously care about that, but you're saying as a neuroscientist, this is what you want to know.
That's right. Don't you want to know what the neurons are doing and who they're connected to? I mean, that's what Mike's talking about.
And is it single neurons or populations?
Hang on to your hat.
And is it response, yes or no?
No, just watch. Okay, just watch. Okay. So here's the strategy that I'm going to develop for you. We have our stimulus ensemble. We probe some unknown network with that. We get all this activity, spiking activity from the multi-electrode. We want to build this manifold in which nearby neurons respond similarly to similar stimuli. Once we have this manifold, we can start to do geometry and topology on that manifold. So we can say, who's related to whom? How are things changing if I move over the manifold? That gives me, underneath this manifold, is a graph that relates the neurons to each other. That's a little bit like an abstract functional network for these neurons. Once I know the structure of this network, especially locally, I can make some inferences about what the circuit properties were that gave rise here. Of course, if I actually knew this circuit, I could derive the manifold, okay?
Why do you make the assumption that nearby neurons are going to be are similar?
I'm building the manifold that way.
Yeah.
I'm building it that way. Yeah.
But then we just heard
It's not an assumption, Barry. Yeah.
mice don't have ocular dominance columns or whatever, right? So that it's salt and pepper.
But this is neurons that are close to each other aren't close anatomically to each other. They're close functionally
Okay,
to each other.
that's.
Yeah. Yeah. They're close functionally. Yes.
Wouldn't that be a matter of correlation of the units rather than the actual network?
Hang on. Hang on. Okay.
So nearby is not nearby, but it's similar.
Nearby neurons. In this abstract. Yeah. Okay, so here's the game, okay? I'm gonna build up the embedding for you, something first, right? So because Mike found that we were getting these responses maybe 2 octaves higher in spatial frequency to the flow patterns than people get to the gratings. Maybe we better look earlier in the visual system to see what's going on. So you brought Greg Field in, who's a great retinal physiologist. He was at Duke. He just recently moved to UCLA. So we're now producing data sets in analogous experimental setups to our ensemble of stimuli and get used to this 6-tuple of arrangements here. We, of course, have grading patterns. We're going to use low frequency and high, and these low ones are right at 0.04, the peak in the Stryker thing before. We're going to do some negative contrast flows, some positive contrast flows. And you're right, we have many other flows we want to be doing, but yeah. The advantage of these, of course, is when we use triples of dots to make little oriented segments, we can titrate the amount of orientation that we have in the flow patterns. There's a lot to be said for that. Okay, if we do this, we have a bunch of these trials for a bunch of these stimuli for each neuron, okay? So we build for V1, here's neuron 1, here's neuron 2. Mike did this experiment many times and we showed that you can combine the information. So we have roughly 1,000 neurons, okay? For each of those 1,000 neurons, we have, here's trial 1, here's trial 2, here's trial 3, over this ensemble of stimuli. So I can't remember the correlation now, but you see that color here goes like that, okay? So each of the clusters in this perceptual manifold corresponds to one of the classes of stimuli in our ensemble, and the individual trials are this cluster of points that you see.
And the axes are what?
The axes are the first 2 significant principal components. We do PCA on this 1,000 neurons and project it down. Okay? So here's V1. You can see right away, Neuralink would have no problem building a classifier to say what the animal was looking at, right? This is trivial. But look at the retina, right? The same thing done in the retina exhibits these classes. The margin between the classes is even bigger, right? So why not just use the retina? What's the cortex doing? There's got to be something more going on. If you look at the dimension of these in terms of, say, the PCA spectrum, you see that the cortex is much higher dimensional than the retina. What's the difference between the cortex and the retina? From the standard perspective, there's effectively no difference.
So the task, you presume a task here, which is classification of
where the animal is doing no task. The animal is simply sitting on the running on the trackball, like you saw in Mike's video, and these frames are coming up with drifting flows of one sort or another, or drifting gratings, second and a half on, half a second off, et cetera, et cetera. There's no task. I'm simply explaining the perceptual manifold by saying, if you want to figure out what the animal was looking at in a particular in some circumstance, you could read that out by building a classifier on this if you wanted to. We don't want to do that, but that's what most people do. That's how they ask, what's the state of the brain? What was the animal looking at? What's the name? Berkeley called that the intent. Yeah, never mind. Okay.
So yeah, a quick question about the temporal aspect here. So these are drifting gratings and the dot flows are all moving?
Everything's drifting the way I showed in that video. Yeah.
Okay. And okay, and then you're just counting total spikes per neuron?
Yeah, for this simple thing here, we're just counting total spikes. If this is the straw man, right? This is we think this is not the right way to be looking at it. Okay. Take a more careful look at how we represent our data. So there's a drifting grating shown to the retina. I show 4 directions of drift here. 1, 2, 3, 4. 4 directions of drift, 4 repetitions. You can see there's a bunch of spikes. So basically, for a retinal ganglion cell, which they have pretty localized receptive field, when the right contrast goes over, boom, you get a burst. When it's not, boom, you get another burst, et cetera, as the wave drifts across. So this should look pretty familiar to you, as here it sees the positive lobe, et cetera. And this particular retinal ganglion cell doesn't care which direction it's drifting in. It only cares whether it's bright or dark as it passes over. So you can see the frequency here. And this is the way we're going to represent the data that says, if I drift in, say, this horizontal direction, here there's excitement, here there's excitement, here it's quiet, here it's quiet. And so you get all those little bumps. So we have direction by time, and that gives us this picture, and color is the rate of firing. Okay? Do the same thing for,
It takes is a significant delay. Oh, you have to reconnect to the Zoom and drop the connection.
yeah, my machine is doing crazy things here. Hold on, it's trying to connect.
Yeah, it's the Wi-Fi is blinking.
Connect again.
Yeah,
Well, you must be connected.
now I'm connected, so I have to reshare the screen.
Yeah.
Okay, so V1, same game, right? Here's Mike's ball with the mouse on it, the drifting gratings, various places. You can see that this is a cortical cell that looks very much like a simple cell. It likes it when the gradient is drifting up or down. So it likes it going like this or like this. And you can see it here. And because it likes both directions, this is a very classical orientationally selective response. So there's big response when the gradient down or up, which means it likes horizontal orientation. And you can see that there are these bumps of response in time, which means it's tuned to a certain spatial frequency. This is all that classic stuff, okay? Now, what happens when things get a little bit more interesting? So that V1 cell that I just showed you gave this response to the drifting low frequency grading. It didn't respond at all to any of the other stimuli. The retinal ganglion cell that I showed you responded to low frequency and high frequency gratings and to positive contrast flow patterns, regardless of whether they were 1 dot or 3 dot, okay? In cortex, here's another neuron, didn't respond at all to the low frequency gratings, gave a beautiful orientationally tuned response to the oriented flows and the negative contrast single dot flow, okay? Anybody know what's going on here? No.
Where do you report from retinal ganglia cells?
Sorry?
Right now, what does it mean recording?
So the way Greg records from retinal ganglion cell is they take a piece of retina out, they put it on an electrode array, a multi-electrode array, which is like a bed of nails, and then they project the image onto that in a very controlled way.
Yes.
Yeah.
And how do you record from the V1 cell? Is that in the detect animal and?
Yeah, that was exactly the procedure that Mike showed in the earlier talk, where the animal has his head fixed, he's running around on the ball, he's looking at these computer screens, and the multi-electrode is inserted in.
And this is in the central part of the visual field? There's 2 neurons or is?
Yeah, these are all from, roughly speaking, near the.
They don't have
There's no fovea.
Yeah, they're in the monocular segment. So it's this part of the visual field that corresponds to the same part of the retina that Greg's Fields recordings are made. And the stimuli are identical. They're calibrated to be, the same magnification, the same contrast. So it's as close to identical as we can make the retinal and the cortical recordings, right? The stimuli are.
So here's our picture. We got this multi-electrode. We got data from different areas. How should we build this manifold in which nearby neurons are functionally similar to one another? Okay. Well, the problem here is, what does it mean to define similarity, right? We're given a bunch of neurons, a bunch of stimuli, and a bunch of responses to those stimuli. Okay. What does similar mean? Well, here's a couple of neurons. Look at neuron I and look at neuron K. You can see to low frequency gratings, they both respond the same. To high frequency gratings, pretty similar. To negative contrast, single dot, negative contrast, triples, no response to the positive contrast. So I and K are 2 neurons. You'd expect them to be nearby on the manifold. And so we need to find some similarity scheme to compute this. Look at I and J, for example. They're okay on the low frequency stuff. The high frequency, J gives a nice response. I doesn't give any response. The single dot negative contrast, I was responding, J wasn't responding, et cetera, et cetera. You can see your way all the way across. So you want I and J to be far apart on the manifold, okay? And so this is the challenge that we had. Yes.
So I'm just curious about these vertical stripes. Is that like an onset effect? Is that the delay?
Oh no. So I didn't really have enough time to develop all that. This is time across here. So sometimes you get transient responses, sometimes you get sustained responses.
And does that mean it's like a certain delay? I mean, is that because the stimulus turns on?
No, stimulus turns on at exactly the same time in every one of these. Okay.
It's like a delay until you get a cortical response, right? So like, is that
Yeah.
once?
Yeah. So you can kind of read the delay right here.
Right.
So if you can see that the time it takes to get to V1, but if you look across here, you see this is delayed with respect to that. So what might that mean? That might mean that's a response due to feedback rather than feed forward, because there's that extra delay. So one of the key parts of the delicacy here was there's a lot of information in these patterns. How do you dimension, how do you reduce one of these response profiles?
Right. And shouldn't delay be or time be imposed as something that you take into account?
We're absolutely taking the time into account. Yes, we're absolutely taking the time into account. Yeah, Shiha.
Conceptually, I know it's not the same, but conceptually, aren't you measuring with a variety of these stimuli something like a receptive field when you have basically noise stimulus, for example?
Yeah, so I showed
each picture, each point in the manifold is like a receptive field, and you're measuring distances between different shapes.
Except the receptive field is different for every different stimulus. That I showed, remember I showed at the beginning what the receptive fields for spike triggered averaging from random noise looked like superimposed on the flow patterns. Those receptive fields didn't make any sense for the flow patterns. right? So we're
figuring out on that because of the nonlinearity.
that's right. That's right. So I want you to move away from thinking about receptor fields and neurons as being filters to neurons being elements in a network, and how do we characterize the functional properties of that network?
But it's difficult to know that you have enough stimuli.
Let's get to that at the end.
You know, there's the concept of receptive field, which is very important for establishing, the features.
Right.
But I think what you're saying is that it was a flawed concept because it was so stimulus.
It's so completely stimulating.
But that's exactly what you expect from a nonlinear network. So that's.
It's not flawed in the small signal regime.
It's
Oh, okay, that's a good point, because a lot of psychophysics is done near threshold.
Yeah.
Where things become linear. It's
right,
true.
right. But
That's a good point with gradings, especially.
Linearness.
But I would like to not get into that argument. Rather,
You're well above thresholds.
yeah, we're well above threshold. I would like you to get into the mood of thinking not of individual neurons being characterized by a filter property, but individual neurons participating in a network. And so we have to understand their function in terms of their role in that network. And that network, like your brain, has to be functional for an ensemble of situations. And we're trying to characterize that ensemble of situations by taking an example of some that are highly artificial, an example of some situations that are a bit more naturalistic, and we're putting them all together. And by working with these 6 tuples, we're trying to look at the multidimensional aspect of the problem all at once. Yeah.
So the population now will be on that.
Yes, the population will tell us about the functional network. The functional network.
So how large are these stimuli in terms of relative to the classical receptive field? Are they?
Oh, they're huge.
Okay, so it goes into the extra.
Yeah. So we're stimulating. That's right. And there's
a big difference.
Oh,
we're 60 by 40 degrees.
Okay. Yes, yeah, they're huge. And it was essential that we be able to stimulate the entire network in order to understand what the neurons are doing. But at the same time, it made the design of the stimuli a real challenge because we had to design the stimuli in a way that we could factor out the positional effects. And so several dimensions are being collapsed in what I'm doing here, one of which, or 2 of which are the spatial coordinates. Yes.
So with the on the left is that is also for your responses, right?
Mm-hmm.
I'm just wondering if the oscillatory responses were out of phase, would you consider it similar or dissimilar?
Oh, yeah.
The phase matters, the position doesn't. And I'm happy to talk to you afterward about how we designed the stimuli to make them positionally invariant. And yeah.
So you gave examples of where if you have a negative contrast or a positive contrast, the same cell gives you out of phase. I mean, it's a phase difference.
There are phase differences. Some of this comes about because the stimuli are drifting in different directions, et cetera, et cetera. Yeah. Okay. So how can we begin to define this similarity kernel? We're going to do it in 2 stages. The first is we're going to express neurons as a multilinear function of their responses, their full response in time to the stimuli. Then we're going to manipulate that so that we can infer a notion of similarity over the neural components here based on the responses to the different stimuli. I'm going to walk through a really simple example of how to do that. But of course, what we're looking for is a way to ultimately get a kernel that we can use for manifold learning. Okay, ultimately, we're looking to get a kernel. Okay, now I'm sorry this whole thing came on at once. So this is a big, complicated slide. Pretend you can't see anything but what I'm talking about. Okay, so here's the game. We have some unknown circuit that we cooked up in the lab. It runs inside this computer. All you know is these neurons are orientationally selective. So we have a bunch of stimuli. We stimulate the circuit with that. We get a bunch of data. Okay, so what is that? We got 200 neurons or 400 neurons. responding to these different stimuli at different orientations for some amount of time. To make it simple, every one of these neurons has exactly the same dynamical behavior, okay? So the first thing that we can do is to say, well, what are we starting from? Well, we're actually starting from a tensor of data. And the structure of the tensor is for each neuron and for each stimulus, there's a full response in time. So think of the tensor as neurons by stimuli by responses, okay? That's the full block of data. There's a bit of work to get all the temporal stuff in here in an appropriate way. I'm not going to talk about the technical details here. But once you realize that the initial data is in this tensor form, the natural thing to do is to say which neurons go with which stimuli for which parts of the responses. That is to say, we're going to do a tensor factorization, a positive tensor factorization, in which we want to be able to relate some group of neurons, like these neurons here, to some portion of the stimulus ensemble with some response profile. Since these are artificial neurons, they'll have exactly the same response in time. And these neurons here happen to be tuned to this stimulus orientation. So the first component of the tensor factorization relates a group of neurons to a group of stimuli with this response. The next one relates a different group of neurons to a different part of the stimulus ensemble with this response profile, ... There's a collection of neurons tuned to 8 orientations. And so 8 factors turn out to suffice for this toy example. So now we have this tensor of data expressed in these components. And the way to read the component is for this loading of neurons and this loading of stimuli, we get this response, okay? So since it's the loading of neurons that we're going to care about, notice this loading here carries implicitly this information with it. So we take this loading here, bring it down here. We take this loading here, bring it down there, and we build up a matrix of the loadings. Now, how should you read that matrix? Well, neuron I participates in factor 1 a lot, in factor 2 much less, a tiny bit in factor 8. Neuron, et cetera, et cetera. So you're saying, well, if I thought of this in PCA-like terms, the columns of this matrix are like the coordinates of a linear space. So that is to say, neuron I looks like, I don't know, this guy here in this space, right? So this is the dimensionality reduction that's of the structure dictated by that. So now in this encoding space, I can think of each neuron in terms of where it is in these neural factor coordinates, okay? And we call this an encoding space because position in that space, position in this space says how close these guys are to each other, okay? Yeah.
But why did you assume, I mean, this analysis in the second line there presumes that the response is the same for everything. Why is that?
Response.
Your response with the neuron.
The response along here.
No.
The
The x-axis on
Above.
Oh, here.
Yeah.
Oh, no, this is an artificial example. It's of course not like this.
Why?
No, we already said these are artificial neurons. We gave them all the same dynamics, okay? And the key point there was I wanted you to be able to think, oh yeah, these neurons are going to be tuned to that orientation. These neurons are going to be tuned to that orientation, etc., without the extra confusion of whether it's transient or, yeah.
Okay.
Sorry, yeah.
What is this word loaded? Are you use loaded?
Loading, yeah. Forget that I said that. The
Terminology for this algorithm?
no, it comes out of the tensor factorization literature. But it's the same thing. If you do PCA, right? So you could say how much does each neuron participate in each of the principal components? That's called a loading.
So how does this relate to SVD?
Sorry?
SVD.
Right, so, yeah, so I don't want to give a lecture on linear algebra, but the game changes completely when you go from two-dimensional matrices to three-dimensional and higher tensors, okay? Much of what we love about SVD and all that in 2D does not happen in 3D. So you have to do a lot of work to make sure these are interesting factors. In the paper, there's 20 pages in the methods section on that. Let me just tell you, all the guarantees that you have, like with PCA, orthogonal, gone. All that stuff is gone. So you have to work hard for that. And we did. That was a year and a half of work. But I'm not going to talk about that. Okay? So
This is not unsupervised, like supervised. You have,
that's all completely unsupervised.
But then how do you get this nice factorization?
Sorry?
How do you get this nice factorization? Like a pattern of neurons just gave with one.
Yeah, so we're using standard tensor factorization packages with some bells and whistles that we've built. And so I want to talk about tensor factorization, because I won't get to the talk.
Yeah, let's go ahead now.
Sorry?
That representation, you need to add some triangles somehow.
No. Let's talk about that afterwards. Okay. Give me the factorization. Give me the factorization. And this structure. So now we have a space. We can define a kernel in that space. And this is another year of effort. So there's an entire paper in Turi's journal on how we build this variable bandwidth kernel. Because the standard ways of building these kernels, where you choose the number of neighbors, like k = 8 nearest neighbors or whatever, can't work if the manifold is not pure, which is not of constant dimension. And yeah, these data manifolds are not necessarily of constant dimension. So these are very non-pure, very complicated manifolds. So you apply a kernel here, you relate each neuron to the other, you build a data graph in which the distance between things is in proportion to the value of the kernel. I'm assuming all you engineers know that stuff. Then we apply diffusion maps, because I'm from Yale, and boom, you get this beautiful manifold. And the manifold that comes out is one-dimensional, and the space in which it's represented here are now these diffusion coordinates, which are related, diffusion coordinates. And you can see that it's one-dimensional, and there's a single latent variable that goes around it, which is the orientation preference. This, of course, is the classic Sokolinski ring model for an orientation hypercolumn. We didn't know that, but that's the way these responses were generated. And you can see that in the ring model, neurons preferring similar orientations support each other. Neurons preferring different orientations inhibit each other. And the manifold of equilibrium in the dynamics is the ring that Turi was talking about. And if you look at the connectivity matrix, if you express it as Turi did yesterday as an adjacency matrix, it's this banded toplets, right? It's exactly what Turi had yesterday. And of course, our algorithm recovers it exactly, okay? So it doesn't recover all the connections, it recovers the functional property that mattered for that manifold. Yeah, Gert.
That's what you mean by this nearby, even though in the actual, it's cortex of the speckle.
Yeah, I don't know where these neurons are in space. I mean, in cortical space. I don't know anything about that, but I know that they're nearby in function in exactly this sense, okay? So are you all vaguely with me? Because now I can start to talk. This is the beginning of the talk. I've got 7 minutes. This is the manifold projected down into the first 3 diffusion coordinates. And what you see when you look at this thing spinning around for the retina, you see these huge clusters of neurons and a lot of stuff, empty space in between them with a few scattered things. Let me play that again, because we love these, okay? So you see this beautiful clustered organization. Look at those clusters.
This is functional space.
This is functional space. Yes, this is functional space. You get these beautiful clusters.
Presumably, each of those clusters has a name in the retina.
Quiet. Quiet. So
Each point is a neuron.
each point is a neuron, remember, and it's easy to say, here's a cluster, here's a cluster, here's a cluster, so on and so forth, okay? So we have all these clusters. Every one of those points is a retinal ganglion cell, okay? What are these clusters? These clusters came about in a totally unsupervised fashion, right? I did a bunch of work, but totally unsupervised, okay? Remember, those are neurons. Since this is a neuron, I know its profile in performance to our ensemble of stimuli, okay? If I look at a neuron that's near that, if I looked at a neuron that was near that, that profile looks virtually identical, right? But you can see, here. So you see, here's two neurons from the same cluster. There's one profile, there's the other, right? See, they're basically identical. But as you move along this manifold, inside the cluster, there's very little change among the response properties, okay? When I walk across the boundary into this nearby cluster, look at those two. They're really different, right? And here's the
nearby.
Sorry?
Why are they nearby if they're so big?
Because three of them, they look the same.
Why is this cluster near that cluster?
Yeah.
I'm going to talk to you about that this afternoon. Don't worry about it. That's beyond the scale of this talk. Yeah. Yeah. If I walk across this low dimensional projection into another cluster, boom, there's a drastic change in the response profile. If I look at another neuron in that retinal ganglion cell in that cluster, same thing, right? Here's the low frequency response, the kind of linear response to what is expected, and this beautiful transient response to the positive contrast flows and the high frequency gradings. The relationships between positive contrast flows and high frequency grading, another talk. Yeah.
I know you're stressed for time, so.
Let me keep going. We'll talk at the end. Okay. So,
Harry and Steve, I just want to say that the retinal people don't use these drifting texture gradients, right? They never use it. And the question is, when you do use it, do you see differences between the classes of ganglion cells that people?
So, if I could shut Terry up for a second and walk you through what he was asking about. If we look at the profile for these neurons with classical techniques, which is using white noise and spatial frequency gradings and a few other bursty patterns that the retinal physiologists love, we discover the neurons in this cluster are on-sustained. That's one of their classes. Here's another class, those are called on-transient. We know that these are physiologically real indicators of a single group of retinal ganglion cells because in the retinal physiology world, they know that each retinal ganglion cell type is distributed over visual space, and they call that a tiling. And so here's the tiling over visual space of our receptive fields for these neurons. But these receptive fields were computed by Greg's lab using classical techniques, okay? So this is a way that we can relate the receptive field structure to this other stuff, even though we didn't use any of those stimuli in getting this. Yes?
So what about the neurons which are in between the clusters? Is it an interpolation between the two?
Yeah, so you have to remember, this is a down projection from higher dimensions. But because of the way Greg sets up his multi-electrode grid, he has a predilection for getting larger cells rather than smaller cells. So it turns out that, let me just jump ahead before I finish answering that question. Greg got great representation from about 10 classic retinal ganglion cell classes. It's thought in mouse there might be many more than that. Some people estimate up to 43 different retinal ganglion cell classes. So we think if he had a better array, some of these would elaborate into additional clusters, and they would be those other classes. That's what we think, okay? So this is a beautiful picture. We basically rediscovered a big part of retinal physiology in a completely unstructured fashion, okay? Go to Mike's stuff, right? What do you think V1's gonna look like?
So just a quick question before you follow. So in this, so on the transaction before is that on transient serves the same functionality. Now you're really determining which cells are similar, and you say, oh, but this actually looks like this.
No, he took, using standard techniques, they were able to classify what those cell types were, independent of our work.
Right.
So we gave him a list of neurons. He said these turn out to be all on transient. We don't know how he did it. Actually, we know how he did it, but he did it down at Duke. We don't have anything to do with it, right? So we were able to infer the classes. He could put the names to it that the field loves. Yeah, we had no names for these, okay? Cortex. Really different, right? Beautiful smooth manifold. Here you're only seeing 3 dimensions, but it's a beautiful smooth manifold. Nothing like the highly clustered structure that you saw in the retinal ganglion cells, okay? The structure of the V1 manifold being smooth says, well, if we walk along it, we have to follow geodesics on that manifold. We're not really walking cluster to cluster and saying, no change, no change, no change, boom, new class, no change, no change, boom. Now we're seeing very gradual changes. So if we start off in this corner of the manifold here, which is this neuron there, you see there's a strong transient response, a bit of sustained response to everything, nicely orientation tuned to a certain class of to negative contrast flows, orientation tuned to high frequency gratings, orientation tuned to positive contrast flows, etc. If we start at this cell, which is this cell here, and we walk across the edge of the manifold here, you see that gradually the response profile changes until when we end up in this corner here, it only likes grading patterns, right? We take this other path across here, you see that you march up, you maintain responses to everything except low frequency gratings, okay? So the manifold can now be colored by the functional distribution of the neurons. And this is a static view of it, that we have lots of other presentations of this, but it's really quite interesting. Over on this side, you see quite promiscuous responses to the ensemble of stimuli. As you move over this way, the responses become more and more specialized. Notice along here, the responses are really just about low frequency gratings, okay? So we think of this as a low frequency arm of the V1 manifold. As an aside to the physiologist, Michael, this is the bulk of the cells that people report from because they're using the low frequency stimuli, okay? They're missing key parts of the manifold. I'm going to go really fast here. We can characterize certain functional properties as you go across here, looking at different diffusion coordinates. There's a beautiful coordinate along the manifold which goes across from negative to positive contrast, right? These organizations were never seen before. Look at this. Mike can take the waveform that he records on the multi-electrode and infer whether the neurons were putative, excitatory, or inhibitory, okay? Look at the distribution. This promiscuous corner of the manifold here are these inhibitory neurons. Remember, he said inhibitory neurons are much less selective than excitatory neurons, right? So this side of the manifold here is all these inhibitory neurons. The excitatory group, look at these guys here. These are all excitatory. That's that low frequency arm of the manifold. There's tons more to be said. Let me just show you one more picture here. We can separate it because he knows where the electrode is. He can guess the layer of these neurons. And so we can characterize whether, for example, look at layer 5, right? The excitatory neurons in layer 5 overlap the inhibitory neurons in layer 5. That's the only layer where that really happens. Look at the excitatory and inhibitory neurons distributions in layer 2-3, right? Layer 2-3, they're largely different populations of cells to each other, very different response characteristics. And so with regard to your comment about columns and orientation tuning before, I think it was you, right? We think this is all about the long-range horizontal connections and the geometric process.
So how did you distinguish excitatory from inhibitory?
Mike did that. It's the waveform.
Okay, the thin spike.
Yeah.
Fast spiking's, yeah,
Okay.
narrow spiking and broad spiking.
Okay. Why would you expect this? Okay. So I'm going to skip over this so I can get to the fun part of the talk. So we love spectral graph theory in my building, and so think about this like you're a spectral graph theorist for a second, right? You have a small tight group of neurons that are richly interconnected. You're going to get a really nice continuous piece of a manifold. If you have a bunch of these that are disconnected from each other, the manifold is going to consist of disconnected pieces. As you start to connect the pieces together in a weak fashion, the clusters are going to start to associate, and the manifold will take on a more clustered-like, but not completely disconnected characterization. And of course, if you add zillions of connections and loops and all that other stuff, the manifold is going to get really continuous, like the V1 manifold. So there's a beautiful spectral theory behind this, okay? Let's go to the opening comment and the reason you all paid the expensive admission price to this talk, okay? Think to yourself, I'm not going to ask, think to yourself, what are these deep networks, right?
They're like the retina.
Hurry. Somebody said to me yesterday, ResNet 50, that's really interesting. It's got these jump connections and everything, right? That's the one that people use. That's the one we all love, right? ResNet 50, okay? 50-ish layers. You all know more about that than I do. It's tuned up to ImageNet, and it does a beautiful classification of ImageNet images into the different classes. You all know that. What will it do to our stimuli? We feed our stimuli into ResNet 50. It gives a response profile across layers that looks remarkably like the typical response profile for ImageNet. And you can see from the classification, it does just fine. It thinks positive contrast flows are spider webs. It really thinks positive contrast flows are spider webs, okay? What do you think the manifold is going to look like? Well, we look at one unit, which is analogous to a neuron, okay? We can evaluate the response profiles exactly as we did before. You see these beautiful orientation tuned responses, et cetera. You see selectivity among the different stimuli. This is the kind of stuff that makes Dan Yeomans famous, right? Because they characterize all this and they say, oh, look, it's just like V4. It's just like IT, because they're looking at a bunch of these, okay? Well, you can look at a single stage. So this is the whole neuron selected by activity. So it's small enough that we can display it. These are the most active ones. That would be analogous to Mike selecting the neurons that are most actively firing. This manifold is even more clustered than the retina.
And this is just the units from the fourth stage. That's the one.
Yeah, this is from the
That's
fourth
pretty
stage.
far away from the input, right?
That's right. And we have lots of stages in the manuscript. Yeah.
Does it have any temporal dynamics?
That's correct. So we invented temporal dynamics by moving the stimulus and building up the temporal response by moving the stimulus across its positional coordinates. So we turned it into a situation that was as close as we could make to be analogous to the recordings that Mike did.
I mean, it's not gonna, there's no, like, frame by frame, there's no, like.
ResNet 50 doesn't have temporal connections, right? Doesn't have temporal connections. So, but that's not what's driving this. What's driving this is that these networks are built with lots of weight sharing and then feed forward organizations. They're a directed acyclic graph. They're like the retina. which goes through those different layers. They're not like the cortex with all the rich feedback from inside and outside all over the place, okay? So you can lay out, this is exactly analogous to the mosaics that I'm showing for the retina, but these are the, quote, receptor fields, unquote, of the units in different layers. And I'm kind of out of time, so let me just wrap up. Oh, there's a couple other layers if you want to see them. Even layer 5, which is the fully connected layer, is still unbelievably clustered. Still unbelievably clustered. Summary. We showed you how to build this neural encoding manifold, and it revealed both a lot of the properties that we understood about cortex, retina, and it inferred those in a completely unsupervised fashion. We discovered that these convolutional networks are actually nothing like, in this global sense, a model for cortex, even simple cortex like V1, but rather they're an even more clustered version of network than the retina. If you calculate the intrinsic dimensionality of this, you get an entirely different perspective on how much richer the cortex is. So we have intrinsic dimensionality is you have to do some work to get it right. The deep networks are really low dimensional. They're only a couple of dimensions. Retina is about half the dimensionality of the cortex. This low frequency arm here, the cells that everybody loves when they record from drifting low frequency gratings or drifting to bores, is the low frequency part of the cortical manifold.
So dimensionality here means dynamical system.
That's correct. Yeah.
They come to dynamics.
Or if you want to think in static manifold terms, you're standing on a neuron in the manifold. How many degrees of freedom do you have to move in or to be influenced by? Okay. Sorry? Yeah. And so we think what's really beautiful about cortex is this leap in dimensionality provides the circuit infrastructure for the emerging properties that we know and love. And that is, my previous career as a geometer was about those properties. So let me just say, this was a spectacular, for me, collaboration, which was this beautiful mixture of theory, cortex, and retina, and physiology and math, and getting it all to work right. If you want to read about the stuff I didn't mention, there's a bioarchive paper about all of this, if you'd like.
That's just one paper, one huge paper.
I'm sorry?
That is one huge paper, I guess.
Oh, yeah. Yeah, I mean, the detail on how to build the multi-scale kernel is in Terry's journal, and that's a 45-page paper.
Okay.
Just a little bit of context. Anyone who's designed any non-trivial chip knows you really can't do it without tools to help you. The standard thing to do if you want to design any kind of chip is you go to the commercial vendors, and if you're at a university, you get the tools for nothing. But if you're at a company, you have to write very large checks, and you get these very expensive tools and use them for your design. Okay. Now, for those of you who are well versed in how these tools work, it turns out to do a really good chip, even synchronous. It's a non-trivial exercise. I'll just leave it at that. And that's what the tools support. Now, when you're trying to design a chip that doesn't use a clock, which is what asynchronous design refers to, this becomes, it's not exactly what the tools are designed to do. And as a result, it's extremely, extremely easy to think your chip is going to work. And then when you get it back, it doesn't. Because the tools aren't designed to check for all the things that you would like to check. By the way, this happens in synchronous chips also, which is why once people have like some, glue and string and scripts that make that, and they fabricate the chip and it works, they never change that glue. Okay, there's a reason for it. Okay. So what we tried to do was we wanted to build a EDA chip design flow for asynchronous circuits from the ground up, or from first principles that has all the right things in it that we need. Okay, and I'm trying to show you what we did. Okay. So, I mean, this is a workshop on neuromorphic computing. So why am I talking about chip design tools here? So the first thing I just want to talk about is, what I mean when I say asynchronous. People use this word all the time for very different things. You can think about computation as being in 4 different buckets: discrete versus continuous value, and discrete versus continuous time. And you can actually populate all of these buckets with well-known circuit techniques. Discrete time, discrete value is digital synchronous logic. That's what you normally learn as an undergraduate in engineering. Discrete time, continuous value, people do this as well. There's no problems with it. Switch capacitor analog circuits kind of fall into this category. Usually you use the clocks to switch various switches and use that to do the computation. Continuous time, continuous value is incredibly popular, like an amplifier. Continuous time, continuous value. But then there is this, neglected quadrant of continuous time but discrete value, and that's what I'm referring to as asynchronous design. So time is continuous, but values are discrete. That's what I'm going to talk about. Okay, and lots of different words are used to describe this. People call this data-driven computing, self-timed computing, asynchronous computing, clockless computing. And depending on who's saying these words, they mean slightly different things, but roughly this is the same class. Okay, and I'm looking at tools for how do you automate design or things like that. So this is a neuromorphic computing workshop. So I just want to tell you that the fact that I'm talking about asynchronous design at this workshop is not random. If you look at neuromorphic systems that are sort of inspired by biology, well, nobody will tell you that you've got this very precise timing signal that runs over your entire brain to coordinate all the activity. So it's a continuous time information processing system. So from the very early days, with the early silicon retinas and cochleas and all of that stuff, it was always assumed that the system has to operate in continuous time. The neuron dynamics and synaptic dynamics are going to be sort of these analog differential equation models that get turned into analog electronics. And the communication between these components is with spikes. And a spike is a very digital thing. So it needs digital continuous time logic. And so if you look at all the early papers, that's kind of how everything is done. And now you start moving into sort of the medium scale. So when the circuit is really small, it's fine. I can just hand design everything. Right? So this is like, everything is hand designed in this world right here. And then the system complexity starts increasing. And people sort of stop using asynchronous design because the commercial, the standard tools that you use to work with this becomes challenging. Okay? And the number of people who understand how to do this in a systematic way is not very large. Okay? It's not a very popular thing to do. So, but what gets very interesting is when you get to very large scale systems, and I'll start with like Neurogrid and even the first Spinnaker, True North, Raindrop, both versions of OE. They all use asynchronous design, and it's because in the neuromorphic systems case, using asynchronous implementation turns out to actually help reduce the energy per operation by a non-trivial factor, like an order of magnitude. Okay, so that's worth it. Okay. And by the way, all the main designers of all of these things, I know them. Okay, it's like you can trace them back to like a small group in one location. Okay.
All in Martin, right?
Yes, definitely. Yeah. My team's in the project. Okay, so the why to use asynchronous logic, but it's mainly for energy efficiency. Okay, so that's kind of what the reason is. There's a biological reason, of course, which was the inspiration for the original project. Like this brain doesn't have a clock running through it, so I want to mimic it. So that's how I have to do it. But there are actually sort of a generic computational reasons as well. So even if you forget about the biology and just ask for an engineering reason, I have a computation I'm trying to do and I want to do it this way. It turns out that if you have extremely sparse activity, the asynchronous circuits give you a much better advantage. It gives you an advantage in power efficiency. And the other thing is you have sparse activity that's not predictable. Okay? So if I can predict the sparse activity, there's a bunch of tricks I can pull. Okay, but a neuromorphic system is a great example of something where the activity is the computation, but if you could predict it, you didn't need to do the calculation at all, right? I already know the answer, so why compute it? So so that's why this is such a good fit for this kind of domain, just from a purely engineering. Now, let's say you could say I could build this large-scale neuromorphic system with a large number of cores with some sort of communication, and I want to do this asynchronously. So now what? Okay. So it turns out that there was really no good answer for this, how to do this. There are many ways people have sort of tried to introduce, I call them hacks for lack of a better word, in commercial tools to try to get them to work with asynchronous circuits. So I'll give you a very simple example of an asynchronous circuit that would make every commercial tool extremely upset. And that's just the ring oscillator. I just have an inverter chain. If you run this through any sort of commercial tool, the first thing it will tell you is you've got a cycle of combination of gates. I'm sorry, I don't know what to do. Right? And that's because in the synchronous model, you've got this hard timing boundary. And you have to cut these cycles at these timing boundaries. Those cycles, next steps in these cycles are controlled by a clock signal that's handled very specially as a separate thing in the design flow. And that's what makes that all the tools are based on that, and that's what makes everything work. And that's a baked in assumption for everything. So as soon as you give it an asynchronous circuit, it's very unhappy. And then there are various hacks people can use. So they will delete some of the connections in your circuit for some of the operations, put them back, delete other. I mean, it gets exciting. I'll just leave that. And even then, they don't actually get the correct answer. Okay, but you can be very conservative and make it less basic. And my favorite, so if you're trying to use asynchronous logic, you're usually out of luck if you're trying to go the commercial route. I love this quote. This is one of the main bullet points and takeaways for all of logic design from one of the standard textbooks. And if you actually do asynchronous design like this textbook describes, I completely agree. Okay? The problem is, of course, this textbook's view of what asynchronous design is actually based on a snapshot in time that's before I was.
And I haven't heard of this textbook. Huh? I haven't heard of this textbook.
You haven't?
No.
Oh, okay. That's a standard textbook for many logic design courses. That's good that you haven't heard of it. So it turns out that there are many compounding problems. So, like with anything that's non-standard, papers don't tell you the whole story. There are a lot of ways you can do it wrong, and so there's not lack of knowledge of good ways to do it. And if you do a chip project and you do it wrong and then it doesn't work, you're never going to do that way ever again. You don't. It's a very painful mistake. And many companies have done this in the past, including pretty much many major semiconductor companies. To the point where, in fact, I remember there's a company that asked me to be, I won't say which, say that multiple. They asked me to be involved in some project and they said, we're going to design asynchronous circuits this way. And I literally just told them it won't work. So I don't want to be involved. And 5 years later, they found out after spending a huge amount of time and resources. Okay, so anyway. But I would argue that a good software infrastructure to design these things automatically cures a lot of these evils because it will impose a discipline on you that prevents you from making these mistakes. So that's what we've been working towards. I'm not going to say we got a great flow because there's no such thing. If you ever need GA tools of any kind, you can always make them better. So this will be a never-ending project, but at least we've got things that we use to actually build it. Okay, so that worked. So that I took a snapshot of a talk I gave at Telluride in 2011, talking about how to do asynchronous design. And it's like, here, how to do it manually. And this, if you do it like this, you can make this all work. Okay, so now that was not a happy state of affairs. By the way, this is the kind of flow we used to actually build TrueNorm. Okay, so that was actually done by hand, and also BrainRock. Terry was involved in that project. So mostly done with a lot of manual effort and a lot of care. Let's just put it that way.
That talk was one of my first introductions to this conference, or this workshop.
Oh, interesting. How do you know?
Here, what have I gotten into?
And now you're here. Right. So today I'm going to talk about the ACT circuit, the toolkit that we've developed. By the way, this is all available on GitHub. There's actually some documentation. I won't claim that the documentation is awesome, but it's actually pretty significant. I use these tools to teach my class on chip design, and all my undergrads use this on a regular basis. So, my group uses this on a regular basis. So, we're not writing CAD tools to write papers. We want to use them to build things. And at this point, I've been using these tools to teach my classes 2018. So every year I use it. It's not a big deal. The students have taped out multiple chips. The first year I taped out like 7 chips, and 2/3 of them work, which is great. That's a pretty good record, by the way, for a student tape out for the first time they've done anything with VLSI. So another thing that we did last year was we actually ran a summer school that talks about all of these things in quite a few details, and actually all of the videos for it are all posted online. You can get off that website, and there's even a Mattermost site that I answer questions. So I get some random indication of who's using the tools because I get questions from out of the blue from people I don't know. So what I thought I would do is I would just start with sort of, if you're used to building chips, I want to start with the key conceptual difference between the way we design asynchronous chips and the way you're probably used to designing digital chips. Okay. And so thinking about designing asynchronous chips using a conventional hardware description language model like Verilog or VHDL turns out to be extremely confusing and massively error-based, which is why we don't use Verilog or VHDL as a digital language. Okay. So don't think about Verilog as a model of how to describe the computation. If you're a software person, this is like super easy, by the way. If you're a chip designer, this is going to be very confusing for you because you have to unlearn a bunch of stuff. Okay. A much better match for an asynchronous chip is a parallel program, not always blocks. So think about the computation as organized in the following way. I break the computation I'm trying to do as a collection of tiny little parallel programs. Each program can receive inputs on input ports, can produce outputs on output ports. And these input ports and output ports are connected to each other through channel communications, and it's a message transfer. Okay, so for example, let's take a pipeline computation as an example that everybody learns how to write this in your favorite either Verilog or VHDL. And instead of doing that, here's how we do the same thing in an asynchronous version of a pipeline. What I would do is say the following. I've got a component that receives an input, does some calculations, and produces an input. I have a second component that does the same thing, the different computations. And then I take the output of one component and feed it as the input to the other. And that's my pipeline. Pretty straightforward. Notice I haven't said anything about how long anything runs, any of that thing. That's going to depend on the complexity of the computation. Okay. All right. But I'm not dictating that. That's the idea. And that's essentially how we build that's the model we use to describe a chip, and that's how we build it. Now, let's see how much time I have. About a little more than half an hour. So I'm going to just give you a quick rundown of the language. In fact, I won't go into the details. I'll make these slides available if you want, but I just show you, I just want to use the tools for most of the rest of the time. Okay, to show you how this works. So the language we use is called communicating hardware processes. It's actually a pretty old language. I mean, we just renamed it. Okay? So we renamed it because the original author was upset at the name. So that we use that name. So it's actually based on Tony Hoare's CSP language, if you know what that is. CSP is communicating sequential processes. So it matches exactly what I said. These components, they communicate, they send messages. It was the first parallel programming language ever in computer science. Okay, so from the origins of parallel programming, more or less. It predates that, it dates back to that. And the syntax is actually based on Edgar Dijkstra's dotted command language, which is even older. That predates, that's sort of the origins of like sequential programming languages with structure. Okay. So it's a very simplified programming. And so, you can assign variables, you can have expressions, you can do A plus B, things of that nature, just like everything else. But it's supposed to capture hardware. So I'm not going to let you do things like allocate memory on the fly, right? Because every piece of state you need to physically realize, so I need to know what I want to do. Okay, and no memory references either. Yes.
So you do have compilers, say, for an SRAM block.
Oh yeah, but that's just an array. An SRAM is just an array, right? And I implement the array with a memory compiler rather than a bunch of registers just for efficiency, right? So, but that's the basic setup, right? And we basically have booleans and unsigned integers as data types. You can specify bit widths. Okay, so that's basically what's in the language. And so I have some more slides on syntax, but let me just, instead of doing that, let me do something just simple here. Let's share my screen. Maybe. Okay, great. Oh, okay, I need to make the font a lot bigger. Oops. There. Okay. All right. So let's write, I don't know, you have a favorite program you want to turn into a chip?
A spiking neuron.
A spiking neuron. Okay, so what are the inputs and outputs that you would like?
Synaptic input.
So synaptic input, so just some number of bits. You accumulate, you compare to a threshold and produce you either produce an output or not.
Anyway, right?
Yeah, so yeah, I sum it. Yeah, exactly. I compare it. If it's not, I just go back to waiting to receive the next input. Okay. Let's do it.
Oh, but this is tricky because there will be continuous integration. How do you do that?
If you want, yeah, I'm just going. This is going to be purely digital event-based calculation. So, yes.
Link the threshold programmable.
Sure. Let's start with something that's not, and then we can make it programmable. Yeah, absolutely.
And we give you 10 minutes.
I don't have that much time, so I'm going to define a component. Okay. We got it. I don't know. I'm going to take an input panel that takes a 32-bit integer, bit width is 32 by default, but we can make it. And then, so all right. So I'm going to have some state to accumulate, right? I'm going to write the CHP for this. I'm going to start it initially 0, and then I'm going to have an infinite loop where I receive a value. This is the syntax I receive. So I receive B, I'm going to do add, and then if the accumulation is greater than or equal to some threshold, I don't know, pick some number, 250 for a 32-bit integer, or that doesn't really matter. Okay, then I produce an output. Else. Okay. First, there's an output, and maybe I say subtract 250. Or you can reset it if you prefer.
Yes, because that would be maybe you should just save them.
You can reset it to 0. Okay. Everybody okay with that as a straw man model? Okay, great.
Maybe the syntax with the full exclamation mark should.
Oh yeah, that's correct. This is thank you. This I'll write it this way. Just send true on output for that. And then this bracket is a conditional expression. So this says if this is true, then do that, else that. Okay, that's basically how it works. And you can have multiple conditions, one after the other. Great. Okay. So there's a bunch of things you can do. So you could simulate this. We actually have a simulator that can execute and run a full simulation. What does the skip actually do? Skip does nothing. Okay. So why is there? Okay, so actually it's a good question. So the semantics of this conditional statement is if all the conditions are false, then you wait. Okay. So you might say, I want to wait for the environment to do something, so you can use the syntax. But here I don't want to wait. So I have to say, I don't want to wait, just quit. Just go ahead.
So else is always manifold.
That's correct. No, it's not always matter.
You wouldn't have the else skips in and then it would wait.
It would get stuck forever here because this is a local variable. But this may be in your port list that somebody else might change. Okay, that starts getting into more tricky examples. And in that case, somebody else might change it, in which case you would wait for that condition to be true before you proceed. Okay, good. So now we can simulate this. So let me do a test example. Okay, so let's say I say test. And it's some sort of test bench. We'll do something silly like send 10, let's say send an infinite sequence of 12s. Output and then sync. Okay. The log here is just for you to see it. Okay. That's and to create the test case, I got it as you got it spiky. And then I can say source. So what happens is the processing just become data types in the language. Okay.
And Rajit,
yeah.
You don't have to save what you receive in the sync, right?
If you don't want the value, you can just say I just say question mark that does the receive and discards the value. But you can put a variable there if you want the value. In this case, you're always sending true, so I don't really need it, so it doesn't. Right. We're going to call it the source. And then it's the sync. Oh, right. That's my task case, right? So I hook them up to each other. Make sense?
Why does Python not get the inputs?
Well, I'm using them there. There are lots of different syntax for wiring things out. Okay, it's not like in Verilog where you have to connect it to ports. I can also do, for example, I can do this. Oh, sync would be really bad. Okay, let's do that. Sync.I is the rest. Oh, I can also say things like this. Okay. So there are lots of different syntaxes you can use for it. Let's see if this works. Made a mistake. Test.act. Most of the code, by the way, the infrastructure is trying to produce useful error messages. Line 27, what happened? I'm missing some grace somewhere. I have an extra grace. That's a good point. Okay. And then I can just run this. It's not going to do it. Run for a while, so step 100. If you get a spike here, okay. That's how it works. And you can do things like I can watch. I can say watch s.I. Okay, and then show you like, okay, I try to send it blocked, then completed. You can see all the events that you want. Okay. And by the way, I can save this as a VCD file if you want. So you can watch it, look at it in a waveform. I'm not going to go into the details of this, but this simulator can also do, mixed. It links against an open source spy simulation called Zeiss, and you can do mixed analog digital simulation with this without any. So.
You still have 10 minutes left.
No, it's good. I want to talk a little bit about the implementation. But I'm just saying we have simulators. It turns out it acts as a functionality so that you can describe circuits at any level of abstraction you want. So you can describe circuits at this level of abstraction. You can describe each gate directly, even with you can go to transistor sizing. All of this can be specified, and this one simulator can simulate all of these different levels. I'll just focus, okay? And you can mix and match them. So you could have part of your circuit at a behavioral level, part at a gate level, and part in analog, and you can just simulate it all together without having to do anything extra.
So how do you manage the process basically now like transistor model in the ACT?
So right now I am not. This is at the behavioral level of abstraction, so right. So all I have is the send and receives and things like that. Nothing else yet. So what I'm going to do next is run this through a tool that will generate a gate level implementation. So far so good. All right. So CHP to PRS. I'm going to say minus B. So this selects a particular style of data path logic, which is like gate combination logic. I'm going to say, please optimize any math you have to do with ABC. For those of you who don't know, ABC is the Berkeley logic synthesis engine, which is got a long, rich history. It's also pretty much integrated with every commercial logic synthesis tool as a part of what they do. And then here, okay, I can give you the ACT file, what I want to synthesize, and then okay, good, always good. So that ran synthesis, and it produced an implementation that's called SDT splicing. Okay, and you can look at this if you want. Doesn't really matter, but this basically ran logic synthesis and generated a gate level implementation. The gate level implementation, and something else it does is it takes the logic expressions that you need, like I had to add 2 integers. Those are all saved here. And what you will see is instantiations from a cell library. So this was converted back into the ACT syntax. This was basically run through the standard combination logic synthesis optimization and imported back into it. That's basically how it is. Anyway, great. So now I have the synthesized design, and I want to now turn it into a chip. Yes.
Is there any chance that this synthesized design would fail, like random delay tests or things like that?
Yes. Plenty of. Great question. So depends on how you synthesize it. Okay. So one of the command line options I gave to the tool was -B that specifies a certain asynchronous circuit design style. In that particular design style, the control logic is robust to any delay changes, uncertainty. Well, not any, but most. But the data path uses standard combination logic, and attached to it is a matched delay. That you have to make sure that the match delay is slower than the combination logic data path. This is local timing system. I could have also synthesized it with a different file that doesn't have that requirement, but anyway, those are the different options. I'm just showing you like one path. One thing I should tell you though is whenever I have an assumption like this that says there's a match delay and that means this signal must be slower than this other one, the ACT language itself can express this as a constraint that the implementation must satisfy for correct operation. And the simulators will check whether the constraint is violated while your simulation is running, for example. And we have some timing tools that will actually check them once you place them arbitrary results. Sorry. So the way we view this is when you run logic synthesis, depending on your circuit family, you may have different types of timing constraints that you need for correct operation. And it is the responsibility of the logic synthesis engine to tell you what those are based on what assumptions it made when it designed the circuit. You don't need to do this in synchronous logic because there's a standard accepted assumption, right? It's set up in real time, everybody assumes that. If you're trying to do latch-based design in synchronous logic, you should see how entertaining that is in the commercial field. I've even heard I won't say who, but one of the major ASIC vendors, their application engineers say, what's a latch-based design? Anyway, so that's a photo of the story. Okay, so I have an implementation, right? And now I want to turn this into a chip, right? I want to play some of this. All right. So to do that, I have to pick a technology. So for ACT, you have to pick a technology configuration file that you can use. That technology configuration file has a number of different details, like what the device models in SPICE are called, all sorts of things. Actually, there's complete documentation on every single config option on the public wiki. Look at it, it explains everything. By the way, we've been using these tools in some shape or form for over 15 years now. So, I mean, yeah, there are lots of things that are there for very good reasons. I can tell you which process technology calls us to add this particular feature. So, great, I have the design, I just now want to run it to places. Okay. And we have we have lots of individual tools you can run, but we've also have them all linked together in a single executable. And we call it interact. Because you interact with the new app. One of my students came up with the name in 2010 as this what he would like to see, so that we didn't have to do so much manual effort. Now it exists. So I'm going to use the Skywater 130 technology because I can install that on my laptop and not get sued. And there's a minor detail, there's a refinement. So it turns out that the ACT language can actually support multiple revisions of your design. And so you can pick which revision you want. Okay, so this is revision 1, which was generated by the synthesis tool. Okay, so now what do I need to do? Well, I should read it in. By the way, that actually helped. There are a lot of commands here, but there's a bunch of stuff here. But I just thought it interact. So I'm going to now read. No, out. I need to read the output. Okay. The next thing I need to do is elaborate the design. Okay, so let's say call it expansion. Because usually in a commercial tool, when you elaborate the design, you flatten the methods. And actually, our tools are written in a way that it never flattens the methods. It turns out, except to interface with commercial. So as a result, for example, I can actually simulate something that's larger than the read on my laptop and it runs quite fast. Everything is hierarchical, except for the state. So I expanded the design. I need to decide what is the top level module I want to synthesize. And I think it was called Spike. Okay. By the way, I could have tried something else. And these things, there's error checking built into a whole lot of commands. So maybe I'm yelling at something. So far, I've only made one type, but that's kind of unusual. But I'm sure I'll make plenty more. Okay. So now I've set the top, and then I want to implement. So how do I turn my gate level implementation into a step, right? So I need to map all the gates into a set of cells. Now, usually when you run logic synthesis, it will pick cells from the cell library. In fact, I showed you the output of the expression optimization like that. But it turns out for asynchronous design, different asynchronous families use different gates. Okay, so depending on your personal favorite circuit family that you would like, and trust me, there are probably 3 for every individual in the asynchronous design community. You might want to do something different. So our implementation flow is actually completely agnostic to what gates you use. I don't care. So the first thing I'm going to say is I want to map this to stuff. Okay. And by the way, I've asked you to map it to cells, but I haven't said there's a cell library. So something what happens. I'm going to say. Okay. So I said map this to cells. And what it said was it ran that. So it walks through the entire design, looked at every single gate in the design, and tried to find the set of gates whose layout you need to be able to run an implementation flow. So it said, by the way, here are the gates that I have to generate. Here are cells you need to implement this particular design. Okay. By the way, I'm doing this with a no cell library. Normally, for a particular logic synthesis flow, you would have a cell library, and then you wouldn't see this message. But if you saw the message, that meant you've got some gates that doesn't happen. Okay, I can see it. Okay, let's take a look at it. And this uses the gate level netlist syntax. So these are the complete definition of all the cells you need. This says there's an OR here, and then an OR in the pull down, and three series transistors in the pull up. These are widths. This is that gate sizing information. Okay, so you can actually go and specify everything you want. You could have manually written this if you wanted. Okay, so you can specify as much or as little of your circuit as you want. Okay, so that's basically how this works. Okay, great. So so let me just do this again. Actually, maybe I should do the following. So I'm going to keep typing the same things over and over again. What did I do? Add, read, after add, expand, add top. ST is sticky. And then it's circuit. But now I have a cell model, right? So by the way, this is how we generate cell libraries. So you give me a logic synthesis. So the way we generate our cell library is we take a collection of benchmarks that we know exercise all pieces of the logic synthesis tool. We run this, and then the file that we just saved, that's my cell library definition. So what I'm going to do now is I'm going to say merge. So when I do this, it won't say I generated new cells because I've already told it by virtue of the fact that I gave it this predefined list of cells. It says, okay, I know you have it, even though we don't right now. Okay. So far so good. But now if you look what has happened, it has mapped all the dates that were in the design have been removed and replaced by instances of cells. Okay. Great. Now I'm going to do this. This generates the transistor level netlist for the entire design. By the way, this at any point I can save a Verilon netlist or save a Spice netlist of my choice. Okay, so if you want to export to other tools. All right. Okay. Now I want to start place and route. Okay. So to do that, I'm going to load in the physical database library. So actually, let me do this here. I'm going to copy this here. So when I mess up, I can replay. Okay, so I loaded the physical database, and I want to create a layout problem for this design. I'm going to do that by saying create a layout problem, and I want to say how much white space to provide. So I'm going to say the cell has some area. I'm going to say, well, let's say I have 1.5 or 1.6 times the area. That's roughly 50–60% density. That's a pretty commonly used number in the place and route world. How do I do this? I can look at the help. So it says create the layout problem and populate the physical database. I need to do the area. I need to do an aspect ratio. So is it square or rectangular? And then for those of you who have had lots of pain and suffering with reading the order of NEF files into your case, if you don't know what I'm talking about, I would be happy. So anyway, so you can say, let's say I say 1.6 is my layout area. Let's make it square, and I'm going to say output.left. Okay. So for those of you who don't know, LEF is a standard library format for physical databases. It's a commercial industry standard that's actually a standard, so you won't get sued for using it. And in it also contains design rules for the router and placer and things of this nature. Okay, so it contains a lot of things in it. And it turns out that the sort of technology configuration information is sufficient to generate what's referred to as the tech LEF. So this is the LEF file you needed for the place and router. And so we auto-generate that. And we generate what you'll see is, let me just show you this output that LEF looks like. Okay, so it's got like all the design rules, layer names, vias, all kinds of things. And then it's got all the macros for all of the cells that it needs in the implementation for, even though I didn't do any layout, right? I don't have any cells. I don't have any installed on my laptop either, by the way. Yes.
How do you know the area you gave was sufficient?
Excellent. So the area I did in VDD create.
Yeah.
You can usually you want to squeeze it as much as possible.
Okay.
But if you squeeze it too much, you'll find that the router will fail, or the placer will fail.
It's just eyeballing.
So you usually, people do various things, binary search, experience, depends on the technology. There are usually good rules of thumb.
Okay.
Also depends how much time you have to spare. So no, because you can actually get pretty good, especially the commercial tools, you can get pretty good packing density, but the router will run for days. Okay. So but if I give it like 5% more area, it might be done in 5 minutes.
Yeah.
I mean, it's like very.
And if you add more metal layers to maybe.
Yeah, but that you don't get to play with, right? Like the technology tells you. So yeah. That's the basic graph. Okay, so okay, I only have 10 minutes left. Let me not go too far. So I created the physical database, right? And we created left, and for left, it needs to know the footprint of your cells, except I didn't do any layout. So it turns out that the layout generator actually creates starting point layout for all the cells we need for implementation as well. And you can save that. And what I will show you is that we generated all of these files. So these are all the cells that are needed for the implementation. And I call them rect files. Why? I've made up this file format, which is a list of rectangles, one per line. So you can parse them with a tiny, whatever favorite shell script you like, scripting language you like. And I mean, I could just have easily have generated a script for, say, a commercial tool, but then you'd be in violation of the license agreement. So this is an agnostic file format that anybody can use, and then probably in half an hour you could write a script to generate skin, for example, or what we do from that. Anyway, so from this you can essentially generate. So I won't talk about this part since I don't have time, but from this you can generate the starting point layout for all the cells. You have to route the cells by hand. Okay. I've done this study for cell libraries and multiple technologies with undergrads. It takes them roughly 1 hour to finish a cell. That's the real thing. And usually you need roughly 50 to 60 cells. So that's what you have. So, but this is the other is one for your technology. Okay, so that's the idea, just like in any basic flow. Once you've done that, then I can run place and run flow. So I've got the thing, I've got initialized the physical database. I think I should just be able to run. Dali is the place, so we just run that. I'm going to turn off the workers. Actually, I told you. Sometimes it doesn't produce any output, people think nothing is happening. So you can turn off the voice. So now I'm going to base design. And I need to give it a target density. So I'll say it's 0.6. 0.5, let's make it. Oh, whoops, sorry, that was a mistake. There isn't enough space. So I give it a target density of 0.5, but I don't have as much space as I need, and so that's an error. We're still working out some things where this should not quit. This was a standalone tool, this should just contain an error, but I can just make sure down in 3. Let's say 0.7. 6 is probably good. 8. Oh, so it ran placement, tells you what the half perimeter wire length is, which is the standard metric all the EDA people care about. That's why it's jeoparding it. It's actually pretty fast. Turns out question, and I'm going to say base IO on the second line. Okay. And then write this to the physical database, and then I'm done. Okay. Now this gives you what's called a placed def. So the def file format is used to exchange layout, and it comes in various versions. You can have it with just the problem of definition. You can have placement information. You can have routing information. It all just extends the same file format. So this is, but at any point I can just say, write def. And I only have a few minutes left, so I'm not going to go through the rest of the flow. I will point you to an example. I have a full documented thing on the wiki. I'll show you that. But just to show you that something actually happened, let's do this with magic. This is going to be slightly entertaining because okay, got it. deaf and deaf, and you have the full case design. And it turns out from those rec files, you can load in all the cells and see the full layout. You can have. Okay.
You have to run this by hand?
No. So this def file, you can just use a commercial router if you want it. That's what we usually do. If your design is small and you don't have serious timing constraints, then you can just do that. If your design is big and you have serious timing constraints, we have a global router that will take the timing into account. And then you can take that global routing result and run it through the final routing stage, so it'll respect that. And then you can use anybody's detail router. That's the idea. Yes.
Community verification also.
So that is a loaded word. So could you explain what you mean?
SQL legacy UVM.
Sorry?
UVM.
Oh, UVM. Oh, okay. Yeah, we are not interfacing with UVM in any way. I could give you a whole other talk on how we actually have a formal verification flow for this flow. So we can actually run a theorem prover to show that the gate level implementation matches the CHP you started from. We actually have tools that do that. I'm still getting them nicely integrated into. So usually the way this works is, a student does a project, does some research, they write some tools. Those tools are enough to write a paper, but not enough to, actually integrate into a flow that you can rely on. But we then migrate that into our flow. So that migration hasn't happened yet. We just worked out the technical details. We have a paper that appears in 2 weeks on how that works. But yeah, so we do have a formal verification flow, but it's much more than just simulation based. So we have something. Yeah.
If you use this high level flow, how good is the outcome compared to if you let someone sit down for a very long time and try to do it?
Correct. Yeah, that's a very good question. So the tool I just showed you, CHP to PRS. So the data path logic is probably better than if you did it by hand, obviously, since it's being run with some automated tools that have been optimized by the synchronous community for 40 years. But the control synthesis is not very efficient. We should hopefully be replacing that, I would hope, by the end of this year. We know what needs to be done. Now I have a student who's actually interested in working on it, so they're writing code. So that needs to be improved. We have a next level, a different level of detail at which you can describe the design, which is data flow. So I don't know, I didn't want to go into that whole flow, but you can actually, instead of starting from CHP, you can start from a slightly lower level of detail, which is basically a data flow graph. We have syntax where you can describe a data flow computation. And we have a flow thread, basically instead of CHP to VRS, use a different tool. And that is just as good as hand design circuits. Okay, so that one. But you're paying, the data flow model comes with some pros and cons, so it may or may not be good enough for your specific application. But if it fits, like if you're doing signal processing, it fits really nicely. You'll get really high quality circuits for that. So it just depends. But the CHP flow needs work. But that I have a student working on that right now. So we should hopefully be able to improve that quite a bit but definitely by the next time. So the other thing I also just wanted to mention, which I haven't shown you here, is you can take either the CHP or the gate level implementation, and we can create a synchronous simulation model. Okay, and so I add a clock, but functionally you get the correct behavior, either of the gate level netlist or of the CHP level description, depending on which one you want to do. And that one you can, for example, prototype on the net. So that's what we do. We want to use the FPGA for fast simulation. So we have a flow that supports that as well.
Yes, so it gave a warning that some cells were need some editing, right?
Yeah.
So these are not boxes.
Correct.
Now, this process, the Skywriter conversion process, has standard cells, right?
Yes.
Can you make use of those? Right?
You can. So, but you will need to add to that library, right? So there will be extra gates you will need, and you'll have to make
Muller stuff.
Yeah, exactly. So there will be a list of gates that you will need to add, and those you will have to do the layout for in the same way as the cell library that you're using. Is designed. Yeah, you can do that. Yeah, people are using stuff.
And you'll be around here for another several days.
Yeah, I'm around on Tuesday, so feel free to bug me. Can show you how it all works. So depends on what you do. Okay, so for example, one part of the logic synthesis is just create combination of novel paths. For that, we actually have an open source standard sample. I will talk about that. It's automatically installed. It's actually based on an older open source library on QiNS Lecture. There is a version of different sound. So it's already written.